National Research and Innovation Agency. A synergy in an orchestra. Established through the Presidential Regulation No. 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by various research institutes, and have more than 70 research centers and supporting units. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
menunggu kita conference room.
Good morning, ladies and gentlemen. I hope that you are well. So before we begin the event, I would like to read the conference guide. So the host, could you please try to share screen the conference guide? All right, so allow me to read the conference guideline. So for presenters and participants are expected to join at least 30 minutes before the conference is started, and all participants are expected to join all sessions in this conference. And please use the virtual background provided by the committee and rename your Zoom username according to the following formats. For presenter, you could use code of abstract or paper underscores name underscores institution. And for non-presenters, we could use the format P underscore name underscore institution. And please make sure to mute the microphone, especially during the presentation of keynote speakers. And in the parallel session, presenters should activate the camera. And questions from all participants are delivered using the raise hand, Q&A, or chat feature in Zoom. And when Q&A or chat feature selected, please deliver questions according to the format name / institution /to name of the speaker and /questions. And every session will be led by a moderator. Those moderator will decide which questions is to be discussed or to be answered. And all participants should fill the list of attendees via the link given in each session and abide all the rules listed on the screen. So now we're waiting for Dr. Iman Hidayat to join us. Thank you.
National Re- National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by various research institutes, and have more than 70 research centers and supporting units. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Ladies and gentlemen, may I have your attention? We would like to initiate the symposium in a few moments. The Honorable Dr. Laksana Trihandoko, Chairman of National Research and Innovation Agency, BRIN, Indonesia. Honorable Dr. Iman Hidayat, Chairman of Research Organization for Life Sciences, BRIN. Dr. Rati Asmananingrum, Head of Research Center for Biotechnology, BRIN. Head of Research Centers and Head of Bureaus in BRIN, distinguished speakers and moderators, representatives from ministries, institutions, and universities, and all distinguished participants. Good morning. It's a great pleasure for me to welcome every one of you to the 8th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering, or ISIBIO 2021, Global Platform on Biodiversity and Biotechnology. ISIBIO 2021 is a continuation of the previous 7 seminars, first held in 2012, and has been annually carried out by the Research Center for Biotechnology, BRIN. This year, the symposium is held in collaboration with Indonesia Biotechnology Consortium, or IBC. The objective of ISIBIO 2021 is to provide a platform for academic dialogue on biodiversity and biotechnology, and for the exchange of recent updates on biodiversity and biotechnological researches. The International Symposium is conducted virtually live from Zoom meeting and official YouTube channel BRIN. Now, before we begin, allow me to read the conference guide. Presenters and participants are expected to join at least 30 minutes before the conference is started. All participants are expected to join all sessions in this conference. And please use the virtual background provided by the committee and rename your Zoom according to the format. For presenter, code of abstract or paper _name_institution. For non-presenters, you could use P_name_institution. And please make sure to mute the microphone, especially during the presentation of keynote speakers. And in parallel session, presenter should activate the camera. Questions from all participants are delivered using raise hand, Q&A, or chat feature in Zoom. And please deliver the questions according to the format, name/institution/2, name of the speaker, and /questions. And every session will be led by a moderator, and all participants should fill the list of attendees via the link and abide all the rules listed on the screen. Ladies and gentlemen, please be prepared and remain seated. We will sing to national anthem, Indonesia Raya. Ladies and gentlemen, we would like to begin the International Symposium with welcome remarks from the Chairman of Organizing Committee ISIBIO 2021. Please welcome Dr. ISIBIO.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. The Honorable Chairman of National Research and Innovation Agency, Dr. Laksana Trihandoko, the Honorable Chairman of Research Institution for Life Science, National Research and Innovation Agency, Dr. Iman Hidayat, the Honorable Director of Research Center for Biotechnology, National Research and Innovation Agency, Dr. Rati Asmananingrum, the Honorable President of Asian Federation on Biotechnology, Professor Dr. Duk J.O., the Honorable President of Indonesian Biotechnology Consortium, Dr. Mastur, all of our invited speakers from Indonesia, Malaysia, Japan, Saudi Arabia, Netherlands, and USA. Also, to our distinguished presenters, participants, and invited guests. Good morning, welcome, and thank you for being here with us today. Ladies and gentlemen, it is my pleasure to welcome all of you at the beginning of these two events. First, the 8th International Symposium of Innovative Bioproduction Indonesia, or ISIBIO 8 2021. And second, the 7th Indonesian Biotechnology Conference, or IBC 7 2021. Today, it marks our first event, ISIBIO 8 2021. It has been 8 joyful years that we host this annual event. And this year is special because ISIBIO 8 2021 is held in conjunction with IBC 7 2021. This would not be possible without a collaboration between Research Center for Biotechnology and Indonesian Biotechnology Consortium. So, in this opportunity, I would also like to acknowledge and appreciate the Indonesian Biotechnology Consortium and the team for making such a great collaboration. Distinguished guests, participants, ladies and gentlemen, this year, ISIBIO 8 2021 has received high appreciation from 107 attendants who represent science, education, and industry communities, including 76 oral presenters, which 65 of them are paper presenters from various institutions and universities in Indonesia and Malaysia. As the output of this symposium, all accepted papers will be published in an international proceeding, IEP conference proceeding, and an international journal indexed by Scopus, Indonesian Journal of Biotechnology. On the third day of the event, IBC 7 2021 will provide you with 2 attractive talk shows. First, we will hear recent updates and the future of biotechnology startup companies in Indonesia from several CEOs of biotech startup companies, including Acumen Research Laboratories, Algae Park, and Nusantix. Secondly, the talk show will discuss program Merdeka Belajar, Kampus Merdeka, in order to share information to us about the opportunity of this national program in the field of biotechnology and biodiversity research. As the chairman of ISIBIO 2021, I would say, please make the best of this opportunity and enjoy casual yet meaningful scientific discussion with the experts on a wide range of topics. Just like the theme of this event, Global Platform on Biotechnology and Biodiversity, we expect that these events can act as a bridge for knowledge and technology sharing, and also an initial platform to spark research collaboration in the field of biotechnology and biodiversity. Last but not least, I would like to acknowledge all the sponsors who support ISIBIO 8 and ISIBIO 7, IBC 7. Tuwadia Prima Mulia, Elo Karsa Utama, Pandu Bioscience, Gitek Jaya, Nusantix, Merck, Crop Life, ITS Science Indonesia, Science World, Gincraft, Biosense Medica, Berkaniaga Medica, and NutriLab Pratama. Thank you for the contribution and support for the events. We hope that these mutual partnerships will continue in the years ahead. To end my remarks, I once again would like to express my gratitude to all of you for your participation and contribution in this symposium. Thank you very much for your kind attention, and please enjoy the events. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Bapak Asep, for the warm welcome remarks. Ladies and gentlemen, next we will hear welcome remarks from the head of Research Center for Biotechnology BRIN. Please welcome Dr. Rati Asmananingrum.
Thank you. Honorable Chairman of Organization for Life Sciences, National Research and Innovation Agency, Dr. Iman Hidayat, Head of Research Center of Organization for Life Sciences, National Research and Innovation Agency, President of Indonesian Consortium on Biotechnology, honorable speakers, honorable moderators, honorable invited guests, distinguished ladies and gentlemen. A very good morning to all of you. It is my great pleasure to address at the opening of the 8th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering 2021 in conjunction with the 7th International Biotechnology Conference 2021. This year, the symposium and conference have the theme The Global Platform on Biodiversity and Biotechnology. The term biodiversity refers to the variety of life on Earth at all its levels, from genes to ecosystem, and can encompass the evolutionary, ecological, and cultural process that sustain life. Biodiversity is very crucial to our life for many reasons, from economic, ecological life support, recreation, cultural, to scientific. Any loss or deterioration in the condition of biodiversity can compromise all of the value and affect human well-being. Distinguished ladies and gentlemen, we are currently experiencing very serious problems related to climate change. It is one of the biggest challenges of our times. It is already happening. Temperatures are rising, drought and wildfire are starting to occur more frequently, rainfall patterns are shifting, glacier and snow are melting, global mean sea level is rising, land degradation is occurring, and biodiversity is decreasing rapidly. All the effects due to climate change have affected two important aspects of our life, food and health. Distinguished ladies and gentlemen, nearly 200 nations reached a climate agreement on COP26 to the role of fossil fuel in the climate crisis. The world needs to cut the rate of the greenhouse gas emission by almost 27 billion metric tons a year in order to limit global warming to 1.5 degrees Celsius by 2030. This effort certainly requires hard work globally and takes quite a long time. We certainly need a strategy on how to adapt and be ready to face climate change. Indonesia is the top 3 of mega biodiversity country, but the problem is that the use of biodiversity is still limited to provide solution to the nation problems, especially to encounter climate change. Distinguished ladies and gentlemen, biotechnology is technology that utilizes biological system, living organism, or part of this to develop or create different products. Research Center for Biotechnology, National Research and Innovation Agency is trying to continue to play an active role in utilizing biodiversity to answer the nation's problem. As a research center that has competence in health, food, and industrial biotechnology, we have carried out various research activities for food security and health. Several research activities for food security are development of stress-resistant and nutrient-rich transgenic rice and soybean, nutrient-rich and long shelf-life cassava, superior seeds cattle, probiotic-based feed for livestock health, and so on. In health security, we are discovering the active compound for medicine from microorganisms, plant, and animal. We also developing the biological based product for infectious and degenerative disease. Distinguished ladies and gentlemen, this seminar and conference is held as an effort for all of us to share our competencies and experiences so that we can collaborate and work together to obtain output and outcome that can answer existing and future problems. We all hope that we can play an active role as part of the world to create a better life. Before I close my remarks, I would like to thank to the organizer committee for their hard work and valuable contribution to this seminar. And my deepest gratitude also goes to the Honorable Head of Organization for Life Sciences, National Research and Innovation Agency, honorable speakers and moderator, the sponsor for their great contribution, Indonesian Consortium on Biotechnology, and all of participants who attend this event. So as a representative of the organizer of the 8th ISIBIO and the 7th IBC, I officially open this event. Welcome to the symposium and conference. Please enjoy the scientific knowledge sharing, global platform on biodiversity and biotechnology. Thank you.
Thank you, Ibu Rati, for the wonderful remarks. Ladies and gentlemen, for the next, we will have a keynote speech by the Chairman of Research Organization for Life Sciences, BRIN, Dr. Iman Hidayat, and which is in this occasion representing the Chairman of BRIN. Good morning, Bapak Iman.
Good morning, Moderator.
May I share
the presentation?
Before that, I would like to read a CV for Bapak Iman. Is that okay?
Yeah.
All right. So before we begin, allow me to read a short profile of Bapak Iman. Dr. Iman Hidayat earned his doctoral degree in plant pathology, Faculty of Agriculture, Chiang Mai University, Thailand. and he is currently the chairman of Research Organization for Life Sciences BRIN, and previously he was the director of Research Center for Biomaterial, Indonesian Institute of Sciences (LIPI). His research interests include development of advanced material technology based on plant lignocellulose and microbial chitin and cellulose. So without further ado, with Mr. Iman Hidayat, the time is yours.
Okay, thank you very much, moderator. I will try to share my PowerPoint first. Good morning, ladies and gentlemen, distinguished participant, keynote speaker, and all participants of the 8th ISIBIO and 7th IBC conference. I hope that all of you always in the good health in current condition and the pandemic COVID-19 condition, so that we cannot meet each other in this great opportunity. But we still can share our knowledge in the current conference. We can contribute to each other about our progress of research. Let me allow me to represent the chairman of National Research and Innovation Agency BRIN, Pak Dr. Laksamana Trihandoko, to deliver policy on global platform on biodiversity and biotechnology research and innovation of BRIN. First of all, I would like to congratulate, of course, to the Research Center for Biotechnology BRIN for the ISIBIO 8 today. And also I would like to congratulate the Consortium of Biotechnology, Indonesian Consortium for Biotechnology, for the 7th IBC. In today's presentation, I will show several policy of BRIN in managing biodiversity, as well as in creating some strategy in biotechnology research and innovation to accelerate the contribution of research and innovation to the national economic development. We all know that Indonesia is a country with a huge biodiversity, from terrestrial to marine to freshwater biodiversity, from animal, plant, microorganism. This huge biodiversity is advantage or basic capital for Indonesia actually to develop economy that very important for Indonesia to develop the economy, because the biodiversity, at least it has economical value, it also ecological value, and also it has cultural value. So it is important for us as a researcher and academic to conserve as well as to utilize the biodiversity. However, in managing biodiversity, it's not easy. Many countries with huge biodiversity belong to developing or poor countries. I don't know why, but because the managing biodiversity is not easy. We, how can I say? We have to maintain, we have to unreveal the biodiversity, we have to develop the technology to utilize. In the same time, the growth of human population also become problematic for, especially for Indonesia, to manage the biodiversity. Growing of population, it means growing in food demand. It means it will need more area for them. It means growing for industrialization. At the same time, the world climate change also increasing. It is, how can I say? It's not easy to managing biodiversity. So in managing biodiversity, at least there are two parts. The first one is how to conserve the biodiversity for future, and then how to utilize the biodiversity for economical development. BRIN has the responsibility in Indonesia to carry out both function, conservation and utilization. For conservation, it is the responsibility of BRIN to provide ex situ conservation. In the ex situ conservation, BRIN provides the biodiversity exploration and inventory program, human resources, as well as supporting funds. And BRIN also provide the facility for the national depository of plant, animal, and microorganisms. And also BRIN responsible for all stakeholders in Indonesia, not only Indonesia, in other countries as well, to access all the biological collection and information preserved in culture collection, herbarium, and museum managed by BRIN. And BRIN also provides services for the identification of biological diversity. For the plant conservation, ex situ plant conservation, BRIN also developed National Botanical Garden. Until now, there are 4 National Botanical Garden, Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Garden, and Bedugul Bali Botanical Garden. Next year, BRIN will develop more national botanical garden, at least 5 more national botanical garden. And in the same time, BRIN collaborate with local government to develop local botanical garden. It is important because with the accelerating of industrialization and also climate change, global climate change, many plants is now going to extinct. It is the responsibility of BRIN how to conserve the plant before extinct. In the utilization, there are also several examples of BRIN to develop a low-level technology and also high-level technology. So back to the biotechnology, there are many definitions, but one of the simple definition is the manipulation of living organisms or their component to produce useful product. So biotechnology is the key for the economical development based on biodiversity resources. It can be for bioenergy, medicine, veterinary, agriculture, fine chemical products, and pharmaceutical. It is challenge for academicians and researchers in Indonesia how we can deliver all of these challenges. Where are we now? We know each other. Where are we now? Do we have this product until now or not? It is the challenge for us. So the current condition We know that biodiversity is theoretically the capital for economical development. And it is the great advantage for Indonesia. And the tool to make it is biotechnology. Unfortunately, until now, we still left behind to other countries, and we has not taken full advantage of this potential. We conduct several biotechnological research and several products such as superior variety of staple food, paddy rice, cassava, and also we do research how to develop superior livestock from local varieties, and also we develop vaccine now, as well as how to develop various vaccine, not only SARS-CoV. But the challenge is not only that, because this biotechnology is the key for food security and health resilience. As Dr. Rati previously mentioned, biotechnology is the key to nation food security and health resilience. But we witness until now, that Indonesian national health resilience is still weak and still not meet the demand. I can show you the case of study for the COVID-19 case. We know that the first case of COVID-19 is in November 2019 in Hubei. The first genome or SARS-CoV was announced in January 2020, and Indonesian first case of COVID in March 2020. Since the first cases, around 6 months from the first cases in Hubei, Sinovac already conducted phase 1 and 2 human clinical trial. And they get the result in June 2020. And Indonesia first administered of COVID-19 vaccine was conducted in January 2021. Until now, already about 46% of Indonesian population has received vaccine of COVID-19. Compare to the Chinese vaccine or Chinese biotechnological research, Merah Putih vaccine have just finished preclinical trial on macaque last month. So we know that there is huge gap between China biotechnological and Indonesian biotechnological. I will show you several bottleneck or constraint in the development of vaccine Merah Putih, in Indonesia. So there are several technology to develop a vaccine, inactivated virus from the viral vector and so on. But the bottleneck is Indonesian researchers and academicians had never developed vaccine. So in terms of experience, skill, we are left behind. The second one, collaboration among researchers, academicians, and industry is also need to be improved, something like that. And the last one is infrastructure. To develop vaccine in Indonesia, we don't have GMP facility. We don't have certificated animal Basel III facility. So it become a great problem for us in the pandemic, how to develop the vaccine. To answer this problem, so BRIN start to develop infrastructure and then how to develop human resources, as well as how to develop the research and innovation environment in Indonesia. Now BRIN developed several facilities in Indonesia, not only in Cibinong, to support the research and technology to utilize biodiversity in Indonesia. This is the building of Indonesian Biodiversity Collection for and data center, and the building will be operated in August next year. And this is the Genomic Biodiversity and Environmental Laboratory building to conduct research on genomics, protein engineering, cell development, cell manipulation, and so on. It will be operated in January 2022. And also BRIN also have BSL3 facility to develop infectious research, disease research. And also BRIN also build the integrated laboratory of bioproduct in Cibinong. All of these facilities are in Cibinong. This not and BRIN also build some facility not only in Cibinong but also in other region in Indonesia, including in West Java, in Sumatra, in Ambon, in Lombok, West Nusa Tenggara, and so on. All are open access for all stakeholders. So not only researcher for BRIN, not only researcher from BRIN that can access all the facility, but other stakeholders, academicians, industry also have the opportunity to access and to utilize the infrastructure through ELSA system. So BRIN developed the system so that all stakeholders can access and can use the facilities. In terms of researcher and industry relationship, BRIN also developed the platform how industry can get involved in developing products. Until now, in Indonesia at least, researcher or research institute in Indonesia doing their research without involving industry. So at the end, mostly the product do not meet the industrial standard or demand. It become chronic problem in Indonesia. Therefore, BRIN now start to involve the industry from the research planning until how can I say, from the planning until the execution, the research involve the industrial resources. So BRIN in this context as a multi-stakeholder collaboration hub, it is not only involving industrial stakeholder but also other stakeholders. To develop human resources, BRIN provides several platforms. For example, visiting researcher assistant researcher, research assistant, and visiting scholar. It is important not only for BRIN, but also for other stakeholders to develop the mutual relationship. For example, in the research assistant, student that conducted research at BRIN has the opportunity to become research assistant and then has the opportunity to continue their study full funded by BRIN through by research program. So it is a great opportunity for students in Indonesia as well as for academicians in Indonesia. Not only that, but also to other stakeholders, so they can increase their competency, knowledge through the system. So BRIN will facilitate research partnership, stimulate multi-stakeholder collaboration, and stimulate the innopreneurship. And we hope that for the next 3 or 5 years, we can harvest the output from this program. I think that's the presentation that I can deliver regarding the policy of BRIN to accelerate biotechnology, research and innovation in Indonesia. Thank you very much.
Thank you, Bapak Iman, for the very insightful speech. Ladies and gentlemen, before continuing to the next agenda, we will have a photo session with all participants. So could you please kindly open your video and the host will help us to take a picture. All right, so there are 7 slides. We will start with the first slides. For the photo, could you please try to give your best smile in this fine morning? Okay, so we'll start for the first slide. 1, 2, 3. The second slide. 1, 2, 3. The third slide. 1, 2, 3. The fourth slide. 1, 2, 3. The 5th slide. 1, 2, 3. The 6th slide. 1, 2, 3. And the last slide. 1, 2, 3. Thank you, ladies and gentlemen, for your participation. Distinguished guests, ladies and gentlemen, we will continue to our next agenda, which is plenary session with speakers Professor Junichi Tanaka from Department of Chemistry, Biology, and Marine Science, University of Ryukyu, Japan, and Dr. Mastur, President of Indonesian Biotechnology Consortium, or KBI. This session will be moderated by Professor Ekowati Hasana. And before we begin, allow me to read a brief profile of Professor Ekowati. Professor Ekowati Hasana achieved a doctoral degree in Food Science Program, Biotechnology, IPB University, and master's degree in Department of Food Science, Nutrition, University of Rhode Island, and for Bachelor, Agriculture Technology, Universitas Gajah Mada. She is currently a professor in Research Center for Marine and Fishery Product Processing and Biotechnology, Ministry of Marine and Fisheries, Indonesia. Professor Ekowati has been conducting researches in the area of marine biotechnology and fisheries product. Now, without further ado, Professor Ekowati, time is yours to lead the session. Good morning, Professor Ekowati.
Okay, yeah. Now, ready. Thank you, Mbak Wiwit. Assalamualaikum warahmatullahi wabarakatuh. Honorable speakers, distinguished guests, and all participants, good morning. Welcome to the plenary session of the 8th ISIBIO, 2021, and in conjunction with the 7th Indonesian Biotechnology Conference, IBC. In the next 1.5 hours, I'll be the host of this session, serving as your moderator. Today, we will be hearing presentation from our speakers, Professor Junichi Tanaka from Japan and Bapak Insinyur Mastur MSc PhD from Indonesia on this very timely subject. But before we get started, please allow me to introduce our speakers. Professor Junichi Tanaka is a professor at the Department of Chemistry, Biology, and Marine Science, University of Ryukyu, Japan. He has been attached with the university since 1996 as instructor and as associate professor in 2007, and finally appointed as professor in the University of Ryukyu since April 2007. He is also alumni of the University of Ryukyu for his master and his bachelor degree. And he achieved his doctoral degree from Pharmaceutical Science, Osaka University. His research interest is on new bioactive compounds from coral reef life, particularly invertebrates. He also engaged in collaborative research with other researchers on related research such as mode of action, structure, activity, relationship, biodiversity, and biosynthetic genes. He has a lot of experience working abroad as a visiting scientist. In 1991, he was visiting scientist at Chulalongkorn University, Thailand, and in 1993 and 1995 at Parmamar, Spain, and in 1997 he was with LIPI Oceanology Indonesia, and in 1998-2007 he was with Max Planck Institute for Biochemistry and Saarland University in Germany in 1998 and 2007. Now, please welcome Professor Junichi Tanaka, who will be present marine natural products and biodiversity of marine organisms. Prof. Tanaka, are you there?
Yes, thank you.
Yes. Your time will be 40 minutes, Professor. Okay, time is yours. Now.
Okay. Thank you very much for the kind introduction, Professor Ecology, Hassana. And okay, let me upload my slide. Okay. Do you hear me? And also, do you can you look at my slide?
Yes.
Okay.
Yes.
Okay. Let me start. Okay. I'd like to thank the organizing committee for inviting me as a plenary lecturer today, and also my former student, Dr. Penny Amadi, for arranging this opportunity. It's a great honor for me. Okay, before talking on the contents, let me introduce where I am based on. if you compare Okinawa from Indonesia, it's so small. Okinawa is located southwest part of Japan and it consists around only 60 islands or so. And it is located east of Taiwan.
Sorry, Prof. Tanaka.
Any problem?
Could you make your voice loud? Could you make your voice louder?
Okay, I got it.
Louder, please.
Is it okay now?
Yes. Yeah, it's better.
Okay, sorry. My voice is usually small. Anyhow, let me continue my talk. Okay. Anyhow, I am from this island, and the island of Okinawa is around. It spans around 500 kilometers in distance. Anyhow, thanks to the warm cross-ocean current in the Pacific, the islands are washed by warm water. So, just like as in Indonesia, the islands are surrounded by coral reefs. Our university is located on one of the largest island, and these are our buildings. And it is located at a nice place to work on chemical and biological aspects of coral reef organisms. Okay. Today, I'd like to talk about marine nature product and some of the diversity work we do. But after the brief introduction on marine nature product chemistry, I'd like to talk about some typical natural product studies, including an example of structural illustration or determination. Some of you may be not familiar with chemical analysis, so please, but it's just part of the basic matter for our field. And we usually look for newer molecules from the marine sources, but even a normal natural product, there is some application. I'd like to show one example. And during natural product study, sometimes you may need to be careful on the research. And I'd like to show one cautious example. As a second topic, I'd like to show some bioresources in nature and also a phenomena of some marine organisms. And so I'd like to show some useful or precious molecules from marine organisms, and also some unique phenomena of metabolites and organisms. And at the last, I'd like to talk about chemical and genetic diversity of marine organisms, of two groups of organisms. These are Sarcophyton, Sophorus, and Iridia mutans. Okay. All right. Natural products are, of course, metabolites produced by organisms. Plants have been utilized from ancient time, but microbiomes metabolites may be utilized from middle 20th century. Compared from then, marine sources, marine organisms are relatively new, maybe utilized after maybe after 1970. One of the pioneer is late Professor Sawyer of the University of Hawaii, and he was dubbed as the father of marine natural products. And why are we attracted by marine organisms? The answers may be the huge number of species in the water. Once it was expected that more than 500,000 species, but some paper reported this estimate, but every year the number is increasing. So I'm not sure the exact number of species. Anyhow, there is a very huge amount of huge number of organisms in the water. Not only sessile marine organisms, but there are also marine microorganisms. And of course, we can expect the environment is totally different from terrestrial area. Apparently, it is aquatic environment. They operate with salinity and pressure, etc. So, after the long evolutionary history of new organisms, they developed different strategies from that of terrestrial organisms. And also there are a number of relationships between organisms. Some are probably parasitic, symbiotic, etc. So, yeah, probably it's endless target. Okay, let me talk about some basics of my method of study with our new example. Okay. First is typical structure determination work, because we are dealing with chemicals, so without knowing what kind of molecules we have, there we cannot proceed further. Okay. So currently, a group of compounds originally reported by Australian workers. And when we worked on a cytotoxic constituent of this sponge in Okinawa, my student isolated two new molecules. These are stereoisomers, or diastereomers to the known ones. One of them, named as Franklin X, is a non-crystalline molecule, but after many trials, my student found that its derivative, having bio in the structure, can be crystallized. And by using X-ray crystallography with diffractometer, you can reduce the whole stereochemistry. With X-ray diffraction study, usually nobody claims that your conclusion is wrong or no. But when you do crystallography, it totally depends on the nature of the molecule, whether it is crystalline or not. In case of, as I told, in case of Franklin X, the original material was not crystalline. And according to Professor Fujita of University of Tokyo in Japan, it is a 100-year-old problem whether the molecule is crystalline or not. Even now, we cannot predict which molecule will crystallize or not. But he invented a so-called crystalline sponge method to solve non-crystalline molecule by crystallography. I heard that currently it can be applied only to small molecules, but hopefully his group invents a molecular scaffold to solve various molecules in future. Yeah, it is developing. Okay, another molecule named Franklin Y was isolated together with Franklin X in the previous slide. In this case, we could not find any crystalline derivatives. So we have to solve the stereochemistry with NMR instrument as well as most conventional natural product studies. But in this case, although it seems to be clear on the spectrum, but there are some signal overlaps, and we face a little bit difficulty on assigning the stereochemistry. So once again, my student try to make several derivatives, and one of them having alcohol at this position instead of double bond. And after doing so-called NOE study and finding out the stereochemistry. Those are the typical works. But in another case, it was not so easy. When we worked on a cultured microalgal metabolites for antiviral screening, we isolated a new molecule named caveolin in relation to a known molecule, protein. Protein was already reported as a cytotoxic constituent, but we also reported its crystallographic structure. But in case of caveolin, not like protein, it was not crystalline. So we used the NMR to illustrate the whole stereochemistry. But with NMR study and additional works, we can only find that 5 partial stereochemical structure colored in differently. So the final molecule must be one of the possible structures. So in this case, I asked my colleague, calculation chemist, to estimate optical rotation value to compare with the actual observed value. So my colleague generated 16 possible structures, and after calculation, he found stable conformals. Then he calculated optical rotation value. And in our final estimate, the whole stereochemistry may be like this, but it is just estimate. In future, I hope synthesis chemists may conclude whether our tentative structure is correct or not. Okay, second topic is the application of a non-natural problem. Polychondramide is a member of trisoxazole macrolide, having 3 consecutive oxazole moiety in the macrolide. This member of molecule was discovered only from marine sources. And they are graphylide, caveolamide, and polychondramide, and microlide, and their congeners. And their mode of action or cytotoxicity or antifungal activity were caused by targeting site actin, the most abundant protein in our body. And with my collaborators, we reported how the macrolide bind to the target protein actin, and also we could solve the whole stereochemistry by crystallography with protein. And we also understood the mode of action. Okay, also the structure is quite complex. We can obtain the pure compound of this molecule from natural sources in gram quantity. And this situation is quite different from some other members, such as capillamide, which are quite complex as a mixture of similar molecules. So we use for some other purpose. In addition, the molecule contains enol moiety, which is a good target for our nucleophilic reagent. So we made several grips based on these molecules. At first, we prepared simple graphic named ISIBIO A and its ephemer by treating icondiamide with ammonia water. By evaluating cytotoxicity against cells, cultural cells, and also affinity to fluorescent GRP, it seems to be that the ISIBIO and ephemer has a higher affinity than original icondiamide. Second application was to make a fluorescent probe. As we know that this class of molecule binds to the leading edge of actin fibers in cells, we plan to stain the peripheral region of cells. By attaching fluorescent moiety through shorter or longer linker moiety, they sustain cells like as shown, as in the right. Okay, the last application was making affinity gel. Nowadays, molecular biology method is used for finding the amino acid sequence of proteins, but I wonder if there might be an opportunity to isolate actin-like proteins. In this case, affinity gel with shorter linker did not work, but with a longer linker moiety, the gel is likely to work. Okay, in the third topic on natural product, I'd like to discuss about artifact formation. When you isolate molecules from natural sources, you may believe that they are natural, but it's not. There are quite a number of possibilities to isolate non-natural molecules, namely artifacts. Here I listed causes to make artifacts. 3 example molecules are shown. The first one is acetone adduct of aldol-like reaction. Second one is hydroquinone. Of course, as you know, it's easy to be oxidized to quinone. And the last one is contains a lactone moiety, but under mild acidic condition, it cleaves into carboxylic acid and alcohol. Okay. So it totally depends on the structure and the conditions when you handle these molecules. In the papers published by my neighbor laboratory, working on my models as well as ours, they reported some molecules with hydroperoxide moiety. These are hydroperoxide. As a hydroperoxide signal in NMR is conspicuous, appearing a chemical shift of 7 to 7.5. So we believe that we did not look over it. However, we have never encountered molecules with this functionality. When we think about the difference between the 2 laboratory conditions, there is a significant difference. Our neighbor laboratory has a big window facing south. That means we are located in the northern hemisphere. Sunlight penetrates through window, and they don't use window shade. In our laboratory, our windows are tree shaded for the southern south part, and the windows are facing north. So we examine the possibility of sunlight. It is already known that singlet oxygen will be formed from normal triplet oxygen, which we breathe, not directly with the light, but indirectly through excited photosynthesis fiber or pigment. As most of extracts from natural sources contain pigments, so it might mean that singlet oxygen may be formed if solution of the material is exposed to sunlight. And in addition, it is a well-known phenomenon that singlet oxygen will react with a molecule containing double bond to give hydrogen oxide through in reaction, as shown in this scheme. Compound 1 is a stable marine natural product from a sponge, and we kept it in a freezer for 3 decades and used it as a model molecule. We incubated in compound 1 with artificial pigment named rose bengal, with or without sunlight. And of course, without sunlight, there's nothing happening. But as it is expected, and if it is exposed to sunlight, hydrogen peroxide, compound 2 was formed. But when we left the mixture for 2 weeks by the window, it resulted in the isolation of further oxidized or degraded molecules 3 to 5. Particularly compound 5 is highly oxidized to incorporate and the peroxide in the molecule, which is a little bit similar to the known anti-malarial drug artemisinin. With conventional oxidizing agent, I believe it is difficult to oxidize position like here. So it might be useful to make some unusual derivative, though the speed, reaction speed is very slow. Okay, the formation of compound 2 and 3 can be explained as in this scheme. The air reaction product 2 will react with water, namely Fock reaction, to give hemichemical compound 6. This compound will flip to give compound 3 tetraaldehyde. But for other products 4 and 5, it is not easy to explain how they are formed. After publishing our results early this year, we noticed that enantiomers of compound 1, 2, and 5 were already reported by high researchers as sponge metabolites. On the other hand, Israel group reported another similar molecule named axiprins as sponge metabolite. I don't know their laboratory conditions, but we have to be careful when you handle the material under sunlight. Okay, I summarize the first part of my talk. When you work on structures of natural products, suitable methods should be chosen for determination or elucidation. We usually neglect or ignore non-natural products, but even non-molecules can be used for some purpose. And because, as I thought, it is better to avoid sunlight to avoid artifact formation. But if you want to have some unusual oxidizing metabolite, singlet oxygen might be useful. Right. For the second part of my talk, Now, in this section, first I'd like to show a few cases of bioactive metabolites from and their sources from Indonesian water. In the latter part, example of the same or similar compound from different organisms are shown. As I worked on some marine resources with my collaborator in Indonesia and Japan, I'd like to show some resource organisms. Jaspermide or just platinolite, one compound has two names because they were different. They were reported by two groups independently. Anyhow, this molecule is a DEPS peptide, so-called DEPS peptide, and showing active polymerization activity. And it is sold as a life science reagent with a good price. This source sponge spread from the Philippines to Indonesia, and you may supply the molecule, though isolation is a little bit difficult. Zesta sponge C is a unique alkaloid molecule and sold as a reagent as an IP3 receptor antagonist. This sponge distributes from Okinawa to Indonesia, so widespread. The problem is purification rates. And there are many similar alkaloids in extract, isomerase or conjugate. It seems to be a very time-consuming job to isolate. Probably due to that tough isolation work, the price is quite expensive. Irimaquinone belongs to melocespherinol. This one disrupts membrane, but there seems to be more biological effect. Isolation is easy. Latranquine A is a macrolide and it binds to actin and has been used for many years. The source sponge distributes from widely from Okinawa, Indonesia, down to Vanuatu, Fiji, at least. You can find this sponge a lot on the underwater wall of Bunaken National Park at Manado, but sporadically at other places. In addition to these molecules, there are more compounds. So you can supply some bioactive molecules to researchers worldwide, and also it may be beneficial to some local researchers. But you need good isolation work together with instruments to confirm the identity and purity. By the way, probably most of the audience are not familiar with sponging. So if you do have an interest, please attend the invited lecture given by Dr. Nico de Bourg early this afternoon, later this afternoon. The latter part of the second section is on the same or similar molecule from different source organisms. Manganese A is a unique alkaloid showing anti-malarial activity. Originally, it was discovered by my former colleague, late Professor Higa, and they coined the name after the collection site Manza in Okinawa. They reported this molecule from two sponges. The above sponge seemed to be quite endemic to Okinawa area, but the other sponge, Acanthostrongyrophora indians, is quite widespread. And I saw this Acanthostrongyrophora sponge in Indonesia at many dive sites as shown in these pictures. From Savan to Sorong and also some down to South Kapa. Some of them do not look the same, but judging from the internal texture or morphology, I believe that they are the same. The chemical contents may be different between colonies. Actually, Professor Tsukamoto of Kumamoto University in Japan and her collaborators at INSURA reported some unique analogs as shown right. Another example is the two sponges. They look quite different, but containing the same molecule but different way. Particularly, copper sponge and microphysicians contain two more molecules named lorimalide and xanthanolide, both of which target microtubules. Therefore, biosynthesis genes are of interest by several research groups. However, it was not reported yet. As we can access both sponges in Okinawa, I supplied 3 specimens each to my collaborator to examine the genetic content. They found that there seemed to be not so much difference on the bacterial fauna for each species, but quite different between species. Also, we expect that the bacterial response for producing methylene can be found easily, maybe through we expected that horizontal gene transfer or same bacteria may live inside, but it was not that easy. Other one is remarkable. In addition to the previous sponge on the left, a different shaped sponge also contains the same molecule, but we have not worked on the right sponge at all. For halcomramide, there is a very similar looking Thai sponge containing the same class of macrolide, namely cabiramide. Also, cabiramide was found from originally from Okinawan migrants. We have not identified which sponge contains the molecule. So, we have not examined the genome of cabiramide sponge yet. Thermal sponges. One of the most studied sponges in natural marine natural flora contain similar compound, namely misakinolide and sphenoide, with or without double bond at this position. Although the outer morphology is a little different, but internal texture are nearly the same. The genes for misakinolide was already sequenced years ago, but one for sphenoide was not reported yet. Although in the literature, cyanobacterial sphenoide was already reported. I heard that the same group worked for misakinolide, also worked on this sphenoide, but so far I have not seen their paper on sphenoide. Anyhow, on the origin of the unique metabolite, there has been a lot of discussion what kind of microbial sources or not. But there are only a handful of results on the biosynthesis genes of sponge metabolite. Okay. Summarize the second and the last part of my talk. We can supply molecules for life science studies from an orange. But there are a lot of unknowns how the molecules are produced in these elements. And the metabolite variation exists even within the same species. Okay. The last part of my talk is on the chemical and genetic diversity of two groups of organisms. First on the soft flora of the genus Phyton, and second on the Piliera nudibranch. Soft flora at shallow waters in coral reefs are easy targets for natural product studies from early stage of marine natural products. And a lot of molecules, mainly diterpenoids and sesquiterpenoids, have been reported. Soft corals of the genus Phyton shaped like mushrooms, and they are rich in membranoid class metabolite, as shown here. About 30 years ago, I was asked to check one of the molecules named Phyto A as a resource in Okinawa, because this molecule was reported to have anti-tumor promoting activity, and someone wanted to develop it. also they quickly soon after we started. And we did a chemical screening on the soft form. As a result, the most dominant species was found to be consist of many chemicals, as shown here. Then we examined further on this group of organs with 3 viewpoints: chemical, morphological, and genetic diversity, together with biologists. As a result, one species, Sulphyton tortioform, collected out of Okinawa Island, is quite uniform in the 3 viewpoints. But the most abundant species, Sulphyton glaucum, was found to be quite complex, and it is likely to be consist of several clades or species as discussed by biologist Professor McFarlane in the United States. At that time, she already reported the species were divided into 6 clades. And more recently, Maloney in the United States and also LC, the collaborator of McFarlane, and our collaborators, including us, reported the chemical and molecular diversity of Parawan sulfite by analyzing mass spectral data of pure compounds and its swap with a platform named GNPS, Global Natural Products Social Molecular Networks, which is available to anyone. As a result, the species Sulphyton glaucum is consisted of mainly 2 clades: blue clade B and orange clade F, in addition to another species, Sulphyton tortioform. With GNPS works, the chemicals can be grouped into the right figure. And each circle represents molecule or related ions. And we may understand that some compounds can be found more in color blue or clade B, and the other in orange or clade F. It is not so clear, but it shows the complex nature of the species. Second group of organs we have interest are nudibranchs, cell-less mollusks, or sea slugs. When we worked on a few species as shown in this slide, we may understand that each species developed a unique chemical for their own details, and they developed it during evolution. And we looked into the most dominant species, most abundant species of the year in Okinawa. By collecting only 16 specimens, we examined their dominant chemicals and molecular phylogeny. As a result, one of the molecule, compound 1, was found from 5 specimens with the same sequence. So we expected that it might be the beginning of the differentiation of the species. To confirm this phenomena, we collected more specimens. After analyzing molecular phylogeny, the species were grouped into 5 species and 6 clades. And with chemical analysis, each clade may express a unique combination of molecules by showing the compound as numbers. But it is not easy to understand how they are complex with this figure. So my student also used the GNPS to analyze the diversity. As a result, the molecules are separated roughly into 3 groups. One is Becquerel type bicyclic compound. Second one is monocyclic or other class of cyclic molecules, and one linear compound. And coloration of each circle indicates a clade. And you can see some compounds can be found only in a few clades. All right, but all for my talks, and I like to summarize. Indonesia has apparently has wide environment, and it's various places and explored places. So, and also as everybody knows, it is located at the center of biodiversity and for triangle. So there must be a lot of bioresources in your water waiting for your research. And for marine nature products, it has passed at least more than 5 decades. But still gives a lot of chances for discovering not only new molecules, but new biological phenomena. And of course, everybody needs motivation to work on each field. It is, in my case, it is fun to observe unique marine organisms in the field and also discover new things in the lab. Okay. Thank you for your attention. That's all my talk.
Okay, thank you very much, Professor Tanaka, for interesting talk. And now we are moving to our next speaker, Tanaka Sensei. Hope you don't mind to wait before Q&A session. Thank you.
Yes.
Okay, our second presenter is Bapak Insinyur Mastur MSc PhD. Currently, Bapak Mastur is a president of Indonesian Biotechnology Consortium and head of research center for biotechnology and agricultural genetic resources. It's under Agency for Agricultural Research and Development, Ministry of Agriculture. He achieved his doctoral degree on bioregulation, Tokyo University in 1999, while his master and bachelor degree was from IPB University, Indonesia, in soil science and conservation, which is obtained in 1994 and 1988, respectively. Bapak Mastur will present in a video form, and please welcome Pak Mastur via video, who will be presenting the current research and biotechnology to enhance crop improvement in Indonesia. Okay, time is for Pak Mastur video.
Everybody. Today I will talk about the current research on biotechnology to enhance crop improvement in Indonesia. Agriculture sector is an important sector in national agricultural development in Indonesia. The sector is very strategic related to food security and agriculture-based economic development. Agricultural development requires support system from human resources, science and technology, genetic, land and water resources, agricultural input and infrastructure, and proper financial and policy system. In this term, agricultural development is directed to achieve advanced, self-sufficient, and modern agriculture. Agricultural development targeted to increase farm production and productivity, land area, and its supported system, production efficiency, which is supported by research, innovation, and mechanization. Indonesia has got target to achieve self-sufficient in several strategic commodities such as rice, maize, soybean, sugarcane, chili, salad, garlic, and cattle toward Indonesia feed the world by 2045. The ambitious target is important and required to set focus and synergy within resources to achieve the goal. However, there are several challenges in future agriculture, including expensive land and labor costs, water scarcity, and expensive costs, decrease on productive land for agricultural practices that force farming system on marginal and suboptimal land, and challenges on climate change impact on biotic and abiotic stress. On the other hand, high demand on healthy food lifestyle and environmental friendly also challenges people who work on agricultural biotechnology to develop crop varieties to meet this demand and challenges. Diversity of plant genetic resources, or PGR, is fundamental resource to develop a new plant variety or crop improvement. There are two basic conditions of agricultural practices that determine sustainable or not sustainable plant genetic resources in the future. A diverse GPGR with normal income condition, development and commercialization of particular local superior variety without conservation activities in the region will lead to non-sustainable agricultural practices that will create biotic and abiotic problems in the future. On the other hand, involvement of in situ conservation by maintaining variation of local varieties in the area will bring sustainable production and commercialization of new varieties by crop improvement in the future. There are several in situ conservation methods that exist in forestry, for example, sagu plantation in forest area, or farming practices in arable area. The other method as a backup in plant conservation in ex situ collection, which applied in the field collection or in the seed bank. Indonesia has several institutes managing ex situ gene bank collection, both in the field and seed bank. ISIBIO collecting 1,000 accession of food crops, for example, rice, maize, soybean, sorghum, mung bean, peanuts, and other edible beans, root and tuber crop. In ISIBIO, we also have microbes collection and insect specimen. Other research institutes will gene bank collection. For example, some Indonesian Research Institute for rice, tuber, and beans, ornamental crop, vegetable, and fruit that cover about 17,000 accession. Plantation crops about 2,000 in total. As my previous statement, the biodiversity is important for crop improvement in the future. Fast development on biotechnology enables new techniques to develop new variety. Conference breeding requires long-time crop and enable molecular marker enhance development of new varieties by molecular marker selection and new breeding techniques with application of genetic engineering by transgenic and genome editing technology. We fit in compatibility on intra or interspecies to create either what we call genetic modified organism or GMO and also non-GMO products. Other progress on in vitro study highlights inter-endosperm culture, protoplast fusion, and in vitro mutation on development of new varieties. Now I will start the topic on in vitro technology. There are three inventions that open pathway for development of in vitro technology. We can say that first invention to cell totipotency-based Swan and Sladen in 1838 was a pioneer for in vitro technology, followed by Harberland in 1902, who culture isolated cell, and invention in vitro germination media by Knudsen in 1921. The current research in vitro technology, especially in ISIBIO, we develop micropropagation techniques for many crops, for instance, in root and tuber crops, horticultural crop, and industrial crop, which is some of them oil palm, sugarcane, bamboo, and some medical plants. Development of new improved superior variety has been released, for instance, on sorghum, rice, citrus, and chili. Application of in vitro technique, for example, in rice breeding, we utilize application of anti-culture to enable development of superior line from double haploid breeds, and combination of in vitro mutagenesis and selection to obtain new rice varieties for saline condition. Some new improved varieties using this method are Bioprima, Biomas, Biosalin N1, and Biosalin 2. Next, I will talk about the sequencing and marker assessment for breeding and crop improvement. PGR including modern cultivar, breeding line, local variety, land races, and crop relative species are very important as input into selection process through plant breeding to develop new and improved varieties. Out of species, small of high cultivated plant species were identified and fruit crops are at loss proportion. Therefore, it is important to set wise sustainable utilization of PGR to achieve food security using advanced technology, especially genomics. The genomic sequencing basically use DNA and also accompanied by RNA for gene expression that could specify a particular protein. All generated data from genome sequencing and further analysis will be for biological sequence of genome tests, and importantly, they are valuable and applicable further analysis. Where do we do genomic sequencing? With this technology, scientists are able to read genetic information. There are three types of genomic. First is comparative genomic. The second, functional genomic, involved in study on functioning and regulation of gene expression and metabolic pathway. And the third is structural genomic, which is determine complete sequence and gene map. Here are some examples of the application of genomics using several factors on crop improvement, such as genomic size, gene number, gene mapping and sequencing, evolution of crop plants, gene cloning, identification of DNA marker that can be developed as marker selection, transgenic breeding, linkage map, and ETL mapping. Importantly, this genome sequencing allows scientists for better prediction and phenotypic character on particular genotype called genome-based breeding. This improve and new varieties and related technology could be beneficial not only for human healthy, also for food safety, but also better environment. One important achievement is establishment of pertanian Indonesia, as can be accessed in this link isibio.litbang.pertanian.go.id, but in copyright in 2018. Genomic information of 11 species is featured the PGPI based on genomic sequencing and genetic mapping. Users are able to look genome browser of some crop with detailed information in genome. This PGPI provides variant for marker useful for DNA fingerprinting analysis and important source of molecular marker and gene governing design traits. Marker assisted selection is main tool for molecular breeding. Especially genetic marker are linked to genetic variant affecting phenotypic change. Second process using molecular marker including single locus or genomic-based marker could be done in seeding phase, so it will reduce phenotypic selection and breeding cycle as well. Middle to high throughputing genotyping is notably for efficiency and reasonable cost and time. And GS is also a proof contribution for rapid development of functional marker as MAS to assist breeding. In case of marker-assisted background selection, this could be high-density molecular to detect UTL. ERJ also developed marker kit to detect early maturity of sugar palm that assist breeding and selection of local accession. In addition, several new varieties have been released and ongoing using marker-assisted selection estimated by Impari HDP, Impari Plus, Impari 40, Biotechnology and as essential variety. Marker-assisted backcross selection have been used for developing Bioni sestigera, which is resistant to brown plant hopper. Molecular breeding also surprise with total species number character higher in biofortification of iron content on rice. And last technique, I will discuss distal genetic engineering, transgenic and genome editing. Transgenic, also known as recombinant DNA technology or gene manipulation, that alter gene in living organism to produce genetic modified organism with a new genotype. This kind of modification are possible inside a foreign gene from one species into another species, genus or higher taxa, altering the existing gene to change the gene expression. Thus change genotype and phenotype, new product or variety. The genetic modification able to overcome sexual barrier incompatibility, and genetic engineering technology can be applied to improve product and environmental friendly. There are three steps genetic engineering research. First, gene cloning, where we can clone gene from unlimited gene source, including from different species, genera or kingdom. Decylated gen modified by molecular by addition of promoter and terminator to be constructed in expression factor. The second is gen transfer to introduce target gen 2. Direct transfer was applied with bombardment particle carbide silica, and indirect transfer was done by agrobacterium tumefaciens factor. The last step component is gen expression, where targeted gen cell be expressed and change the intended phenotypic character. There are several national institutions working on genetic engineering to produce GMO. For example, ISIBIO has been developed Biogranular agriculture, GMO potato resistant to Phytophthora infestan. Developed by collaboration ISIBIO and Cornell University. Also, drought research sugarcane already released by PTPN9. Other research are developed by insertion of alanine aminotransferase gene driven by root-specific promoter OSIRE. And the current status of this study in confined field trial to select baseline. There's also tomato GMO for CMP and TYLCV virus resistant, which is currently on multiple trial CVT for food and environmental safety. The next technology is genome editing. This technology modifies gene or genome by inserting, replacing, deleting, and transferring precisely with the help to an endonuclease Cas9 enzyme as molecular scissor on specific target. Some example genome editing system are GFN, Talent, and CRISPR-Cas9. CRISPR-Cas9 currently is most widely used and preferred because the system is more effective and editing application, drug discovery, and gene therapy to cure genetic disease. A pipeline for using CRISPR-Cas9 mediated genome editing to generate plant knockout mutant started by CRISPR-Cas9 construction and interrogated plant or known as plant transformation. Screening and regeneration of selected transformation were assessed, followed by genetic and phenotypic analysis on transformation plant. Genome editing system has been concerned in some university and research institute in Indonesia, not only ISIBIO, but also Gajah Mada University, Bandung Technology Institute, and also Indonesia Research Institute for Biotechnology and Bioengineering. ISIBIO already worked on a CRISPR-Cas9 genome editing system since 2016. Research objective to obtain rice improved variety for semi-dwarf character and potassium efficiency. There are also CPVD virus-resistant citrus, increasing artemisinin content and chili resistant to Gemini virus. The CRISPR-Cas9 genome editing research from Gajah to get albino or seed by mutation BDS3 achievement. Ministry of Agriculture, University such as ISIBIO, UGM, UNED, and research institutes such as LIPI also have been conducting research in biotechnology. Indonesia Institute of Science also obtained some new crop variety from clonal variation, marker resistant, irradiation, or genetic engineering technology. Biotechnology has contributed a lot on crops improvement and new varieties released to meet national program or human need and environmental friendly in general. ISIBIO has been released some improved varieties. From example, from marker selection, we released 5 rice and 1 chili improved variety. In vitro technology become a great tool to produce 4 rice, 1 chili, 1 tangerine, and 3 sorghum new improved varieties. Regarding modern biotechnology research, Indonesia has released already drought-resistant sugarcane by UNED and Phytophthora infestan-resistant potato by ISIBIO. And from latest technology, CRISPR-Cas9 genome. That's all my presentation. Thank you very much.
Thank you, Pak Master, for a nice video talk. Okay, ladies and gentlemen, question for Bapak Mastur could be written in chat, and the committee will send your question directly to Dr. Mastur by an email. And I'm sorry about this. You can wait for the answer after the committee send an email to Dr. Mastur. Okay, so now we back to Professor Tanaka. Tanaka Sensei, are you there still?
Yes, I am.
Okay, to remind us on the lecture from Professor Tanaka, it was very, very interesting, Sensei. There were three topics actually, and interesting one is the Professor Tanaka closing with remark that it is a big chance still for Indonesia to find new compound in the lab due to the unique location of Indonesia marine. Okay, so in chat we have 4 questions, Sensei. Yes. I will read. I will read them. Yes, the first one is from Ibu Yakti, Pusat Studi Biotechnology UGM. I just wondering how you realize that you identified the new structure that have a potential to be developed in the future. Okay, maybe one by one, Sensei. Okay. Or...
Yeah, of course. I just replied short on the message, but I'm going to answer directly. Of course, when you want to develop the molecule into some other application or something, in that case, the original molecule is expected to have some biological activity usually. In case of, for example, halichondramide, it targets G-actin in our cells, and or just showing some toxic activity or antifungal activity. But in addition to that, it might be useful, for example, by attaching some kind of antibody. There is so-called drug group named antibody drug conjugate in ADC in short. So because of that, there is some easy to attaching point in the molecule structure. It might be useful if the molecule can be supplied in a synthetic biology method or recombinant biotechnology technique. But it's not it's a totally different field from my own. But it's not easy to supply. We better not think that we supply the molecule from nature, but by technology technique. Is that all right?
Yeah. Okay. I forward to number 2. It's from Ibu Tuti Murniasih. It's in Ancol. I think you visited there already.
Maybe Tuti is the one I met already many years ago.
Okay, yes. His question is, for compounds that have been applied as life science reagent, it is produced in synthesis process. Is there any most effective strategy for the selection of compounds from the isolation until applied?
Okay.
Sensei, we will have time until 11:28, and we still have 3 questions. So please, be short and
Okay. Let me do. One example is that so-called domoic acid or kainic acid. Once a simple molecule like kainic acid, it has been supplied from nature. But nowadays, I believe some synthesis people made a strategy, so it is supplied by synthetically in part. And also, but in the actual drug, like so-called eribulin, it is against breast cancer, and this drug is quite complex in structure. But synthetic people also produce the molecule because the in this case medicine is quite expensive, so you can it will be there is a reward for supplying by synthetic. So it is not simply on the structure, just by the structure, but the values of the molecule.
Okay.
Is that all right?
Can I read the third one?
Yep.
Yes. Okay. It's the question is from Ibu Tika. It's a researcher from BRIN. Thank you, Professor Tanaka, for the nice and interesting presentation. I have a question with regards to the diversity of marine secondary metabolites or bioactive compound. Are there any influence on specificity in the taxonomy, either species or family of respective organism, or do you think it was rather affected more by geographical or environmental factors or microbial simion? Thank you.
Okay. Judging from the molecular diversity, microbial resources, and also some among the cells-time organ sponges are the most prolific sources of various molecules. In case of, for example, corals, soft corals, and also stony corals, they are not. They provide a huge number of terpenoids, but from the structural diversity, they are not really exciting. Okay, and what is the answer? Geographic factors, microbiome. It's not that easy. We cannot answer easily. There are many cases, and in some, it was already known that some organs are quite different between colonies. And in other case, most any colony contains almost the same molecule. So, and there are, of course, some people examine microbiomes in bio, but it's not easy to conclude as a fauna, a microbial fauna in this kind of organism. As you can see in some of the literature, after obtaining so-called metagenomic DNA from sponges, you can find many groups of bacteria.
Okay, the last one is from Pak ISIBIO. It's about modification of the structure, the functional groups, or modifying the elongation of carbon bond. Are there any general rule or about steps that we have to consider first for modifying the compound structure? And the second one is, a huge marine biodiversity exhibits a huge diversity of marine-based biochemical as well. So what is the current strategy in order to accelerate the discovery and development of drugs from marine organisms?
Okay.
So,
for the first question, in case of for some derivatives targeting protein, because of the huge size of the protein of the compared from the so-called ligand molecule, like natural product, probably we may need some we may need to attach some linker moiety, but I don't have much experience on this aspect, and it is better to consult other research papers when they target fish the target protein from with the natural product. And for the second question.
Yeah, for the second question.
Yeah. Nowadays, big
about
pharmaceutical companies do not prepare a natural product library by themselves, but small venture-like company prepare. What I heard is we can provide library of natural products, and these companies screen against for some drug target. And find out the hit, I believe.
Okay, that's all, I think. If you have any question more, I think can we write an email to you, Prof. Tanaka?
What?
If the audience have any more questions, because we have a limited with the time. So can we send an email to you?
Oh, no problem.
Okay, thank you very much. Okay, thank you very much for Professor Tanaka and
it's my honor.
Dr. Master for addressing very interesting topics. I won't take a summary because it's very, very deep and well, I think it's very, very broad, also an interesting topic, and I think a lot of our researchers here is eager to actually make communication with you. Yeah. Okay, on behalf of the committee, I would like to thank to all speakers and to participants to the active participation. Thank you for joining us today, Sensei, and also for all of you participants. Now, I'm giving back the time to the MC. Yeah, I think the time is up and exactly, okay, thank you, Mbak Wibit. Thank you, Professor Tanaka and
Thank you. You are welcome.
Giving applause, big applause to the. Thank you.
Thank you.
Thank you, Professor Ecology, and also Professor Tanaka and Dr. Master for a very insightful session. Distinguished guests, ladies and gentlemen, we would like to remind you to please kindly fill out the attendance list of the first session that has been informed in the chat feature. Now, for the next agenda is Principle and Mechanism of Biotechnology Product Session. We will hear presentation from Mr. Alfian Mauludin from Tech Jaya. Mr. Alfian Mauludin, you would have 30 minutes and the time is yours.
Okay, thank you. Let me share my presentation first.
Sure.
Is it already up here?
Yes, it is up here already.
Okay.
The time is yours, Mr. Alfian.
Thank you. Okay. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. I'm very pleased to see you all. Thank you, and thank you for coming to this presentation. And really thank you to for give me this kindness of opportunity so I can be in here. Before I start my presentation and our discussion in this opportunity, let me explain to you and give the company profile for Detect Jaya. Maybe I share first.
Excuse me, Mr. Alfian. The sound, we
Sorry?
cannot hear the sound. Maybe.
Oh yeah, sorry.
Sorry.
I
Also the
will
sounds with the video.
share the sound. Okay. So.
Okay.
Okay, thank you for your listening. Thank you for your attention to see my company profile video. Before that, allow me to introduce myself. My name is Alfian Mauludin. I'm from sales engineer PT Detect Jaya. As you know before, PT Detect Jaya as a solo agent Shimadzu Indonesia. Okay, in this opportunity, I will share to you all about the analytical intelligence with Shimadzu solution. What is the analytical intelligence and what the Shimadzu solution for facing the next future maybe inquiry should be in the next future. Okay. Like we know, there are some difficulties in operation, such as diversification of working. Right? There are two types of diversification of working. The first is work efficiency, and then the second is variety user. Okay, as we know, day by day, the challenges in work are always increased, right? Such as the challenges for the increased job, many job or many tasks with a limited time. So what we need in here is how the instrument, how the technology can be support us to give them good efficiency or operator efficiency, right? And then for the variety user, there are two type skill operator. Is it like export operator? Okay, maybe no problem or no issue for the skilled operator or skill export operator, but how the beginner? For the beginner, maybe need the more technology or more help to that the task for this. How make everyone, even beginner user, can use it the instrument as easy as they can. Okay, then What the Shimatsu want to introduce to you all is it the analytical intelligence. Analytical intelligence in here means a smart analytical platform that incorporates state-of-the-art EOT. Is it like Internet of Things, or everything about internet, everything about networking in here, and then analytical intelligence and machine learning algorithm functionality. So is it like machine we test machine to learning the algorithm function so that the machine can helpful to make our job, our task in laboratory especially to be easy. Okay, and then the analytical intelligence means to automation workflow include auto monitoring, auto controlling, auto diagnostic, and auto recovery function release the user from routine instrument operators. So when instrument have problem, instrument cannot be what I can say can be recovery without our help, something like that. And then what the purpose for analytical intelligence? Is it like for minimizing system downtime and then optimizing efficiency? When we get the minimizing system downtime and optimizing efficiency, so the productivity, our productivity will be increased. We will get the maximum productivity in our activity, especially in laboratory. Okay, next. What instrument have implemented this analytical intelligence? Okay, the first of thing is ultra high performance chromatograph. Is it like UHPLC, right? And then as you see in my slide, in my presentation, is it the newest UHPLC from Shimatsu? We call it Nexera series. It means like next era or next generation era for facing any diversification of the working. Okay, this is type is it actually for modular system, so very flexibility for upgradable and as your own configuration and as your need in the laboratory. Okay, then. So what the intelligence in our HPLC, maybe you can see in here, this is like a routine workflow, which we always do in the laboratory, right? The first time we start up and then we do set up mobile phase. Is it like a important thing when we're using chromatography, and then we do column equilibrium, or we waiting until the system stability. So after system stabilize, we do analysis, start analysis. We inject a sample or standard in there. When the system running, we need to check, is it mobile phase check or is it enough or not? After that, we get a result and then we do system monitoring and a shutdown. And then we will using the instrument in next project or in another day. Okay, so how about the intelligent workflow? Actually, the intelligent workflow like I showing in here, we can see every manual process can be run as automatically processed, such as auto startup. We can set instrument and accordingly what time we set the instrument will be turned on even we not yet arrive at the laboratory. Is it possible to do that? And then do mobile phase setup. So for mobile phase setup in here, we have a technology intelligence in here. We call it advanced real-time mobile phase monitoring. What is the advanced real-time mobile phase monitoring? Is it actually this feature for mobile phase setup? We have real-time mobile phase monitoring in here. We know the important things in HPLC. Some of them is mobile phase, right? When we not care about the capacity of mobile phase, it can be problem for our instrument and for our analysis. Actually, in here for mobile phase monitoring, is it like a sensor? Maybe I will show you in here. This is the sensor in here, gravity sensor. So when you put the mobile phase bottle in here, content mobile phase sure in here, so the system will be calculate automatically can be calculated automatically how much volume inside the bottle, right? Then we can set up the minimum volume for the mobile phase in here. So when the system decrease until the minimum volume which already we set, this it will be send the notification in case of shortage. So the mobile phase time monitoring can be installed and can be directly using in our smart gadget, our smart device, like maybe smartphone or something like that. And then what the purpose for advanced real-time mobile phase monitoring? Sure, is it for prevent system dry up? Is it like when we using the instrumentation and the mobile phase to be dry up? Is it will be impact to our column? So column will be easy to damage, and then another that the result or the analysis will be not good as well. So we need to reanalysis, so wasting time for this. So real mobile phase monitoring, is it means maximum of time. Okay, this is for one patent from Shimadzu for analytical intelligence. Is it advanced real-time mobile phase monitoring? Okay, then the next, after we make sure for mobile phase, then we do monitoring or column equilibrium in here until the column stable, until the system stable before we inject a sample or standard. For the conventional method, we do conditioning column with plusing instrument with set up flow rate. Example like this, maybe we set up maybe 1 milli per minute or more, something like that. When we first turn on the instrument, turn on the pump, will be make a pressure jump in here. If you using the higher flow rate, right, and then pressure jump in here will be impact to your lifetime column. Even that, so Shimadzu released analytical intelligence technology for flow pilot. Actually, flow pilot is it run the mobile phase flow rate gradually to set the point automatically. Is it mean when we set up 1 milli per minute in here, the system automatic run in a half of flow rate? Is it like 0.5 milli per minute in here, the example, and then when the temperature increase gradually, the flow rate will increase too. So this is prevent the extra pressure, so will be save your column lifetime. Okay. Another thing from the flow pilot protect column, is it everything automatically. You just set up in the software, and the software will be start your method, and then running the flow pilot protect column technology here. Okay. Then after we get the system stabilized, and then the column equilibrium, we got the good stabilization in there, we starting to start analysis, right? And then after, when we starting the analysis, we need to check the mobile phase, is it okay or not, enough or not, something like that, and then do system check. Maybe sometimes we get no good result, when we do analysis. Is it can be because is it because maybe from the instrument, or maybe from our sample itself? Okay, so in the Shimadzu, they have another analytical intelligence. We call it auto diagnostic and auto recovery. It's mean like instrument can diagnostic if there are something problem in the instrument, but they can recovery too by themselves. Okay, so no need our help to recovery the instrument. Okay, what's the meaning in this slide is like this. When this normal pressure, right, when we do analysis in here, we inject, okay, in normal pressure, we will get the good result in here. But when sometimes the instrument or the pump, there are a bubble maybe in the mobile phase, so the pressure will jump the pressure will drop. So make a fluctuation in here. Pressure will be fluctuation. So when we inject in this condition, you will get no good result from your analysis, because that after we on this feature auto diagnostic, instrument will do auto purge to prevent this abnormal pressure. After auto purge, and the automatically system will give the normal pressure to normal pressure, and then conditioning again. After everything okay, system automatically will be re-inject your sample. So if the failure in the sample too, and then auto purge happen in here, the system will be re-inject sample to before. So this data will destroy and not saved in the database. So you will get the great result without you must waiting in the laboratory. So the benefit from this, yes, the chemical does not need to be physically present when abnormal condition appears. So after you preparation, after you start injection, you can go out, you can do another job, another task, then you will get the result even in there. Nextera series can perform auto correction, ensure maximum system uptimes. Okay. Okay, and then after we do auto diagnostic and recovery, we got the result, and then we are reporting. So we calculate the result. We need to make sure if our instrument have a good maintenance schedule. So the Symposium solution give a smart service net. It means like some feature in our software, some feature in our chromatography software, like give the approximate maintenance timing by prediction from the use history. It's mean like when you must calibrate, when you must replace another part of like a consumable part or even spare part in there. You will not missing for the maintenance your instrument. If your instrument highly good maintenance, I think the lifetime is good to write, and then your result will be great. Okay. Is it that's all the simplest and intelligent workflow from Shimatsu? Is it our intelligent workflow automatically intelligent workflow in here? So important and so helpful for us in the next project. Okay. And the next slide, I want to introduce to you the newest gas chromatography. We already talking about liquid chromatography, right? Okay, so I want to share with you all about the gas chromatography. We call it next industrial standard GC generation. We introduce to you about a new experience, intuitive and simple controller like a touchscreen, GUI, graphic user interface, and then what the meaning graphic user interface is mean like what the display in the GC instrument is it similar or same in the software too. So easy to make so easy to use this GC. And then we have new graphical icon improve operability. It's mean like the easy friendly use for your laboratory activity. And then we have innovative software enhancement like quick batch. Quick batch mean like when you are watching a movie in studio, the software will info you what the number file empty and already fill up, and then you cannot fill again if already taken by another file. Similar like that. And then we have lab solution direct. Is it enable remote control and monitoring? Yeah, is it really useful when you're using when you working like a work from home? You can direct access. You can connect to your smart gadget, something like a smartphone or tablet. Is it another tools can be connected in there? And then the important thing in our new GC is tool-free routine maintenance. like I introduced you before, there are some talking about variety user rate. There are two type user. Is it a beginner and expert? This new analytical intelligence from Shimadzu purpose will have the beginner user to make everything easy. First, we need click tech nut. Is it like for replacing like a septum or glass insert? Maybe you can see in this tutorial video to make it easy to be understanding. There is just only need 30 seconds to replace stone or glass insert. We can compare with a previous technology. We need tools in there, so need time to replace it. So you can so the instrument get the other benefit is that save your time. And then how about the click tech connector in here? Click tech connector in here means when you install new column, so you no need anything tools be used in here. Just only click tech connector, just open the GC and then you plug and play to the injector port. Something like this, so easy to beginner user. Like this, and then we have a smart jig. Smart jig, it's mean like a jig for make sure everything already according a GC requirement. Depend the detector you use it and the type of column you use it in the gas chromatography. So you need jig to measure how long the column installed to the injector port or to the column port. To the detector port. And then the simplest thing in new GC Shimadzu technology is it like a breader oven. Breader oven, maybe some people know no notice for the lights we need inside the GC to get us when we do installation or when we do maintenance in the GC. Okay, Shimadzu already have a breader oven inside the GC column oven. We have to clean gas flow line in here. The point is we just need a plug and play the filter when we do installation or when we replace a new filter in here. Okay, as you see in my presentation, you freely create your own configuration. Shimadzu give the flexibility for your configuration. You want to you want to create your own configuration, like such as you want to add with headspace system auto sampler, or you want to connect with thermal desorption or pyrolyzer or something like that. And actually, some of this configuration already installed in Brain. Or ISIBIO or another city. So in this slide, let me introduce to you all another product from Detect Jaya products. We can over to you. We can give your solution for your inquiry in the laboratory activity, maybe in the simplest like a spectrophotometer. We have a molecular spectrophotometer, UV-Vis, even like from the double beam spectrophotometer until a single beam spectrophotometer. And then we have a Fourier transfer infrared, or we call it FTIR. You can see in here like a portable FTIR. You can move the FTIR to another place. Is it ER Spirit? And then we have a Huygens ER. Is it ER tracer can be upgraded until near. Okay, then we have spectrophotometer atomic. We have ACE. ACE is from like elemental analysis, tracer analysis. You can use it to measure elemental analysis from percent and up to the ppm. And then we have ICP and an ICP-MS. Is it more sensitivity for spectrophotometer atomic, which can measure until PPT levels. And then we another just we have a chromatography HPLC and GC, and then we have mass spectrophotometry such as like LCMS, single quad or triple quad, LCMS MS, and then we have GCMS single quad and GCMS triple quad too. And not forget, we have another solution for your unknown analysis. Is it your QTOF MS MS from Shimatsu, which can combine with imaging imaging system IMScope. Is it in the process installation in LIPI? And then we have another product like a thermal analyzer, like differential scanning colorimeter, and then TGA. We have another solution like for MALDI. Is it already have in LIPI2 in brain performance MALDI TOF in there. So we have a newest MALDI like a MALDI Mini and MALDI 8020 for the benchtop MALDI. We have another instrument like total organic compound for your environmental analysis. Is it like a simple instrument? Another simple instrument like balance, analytical balance, or top loading. Okay. The next is it like I want to introduce to you all. Beside the instrumentation, we have another solution for your laboratory environment. We call it a software solution. The challenges from this inquiry from every single laboratory is how to integrate the process with instrument become one solution. Is it we have a solution using a software. We have lab solution CS or client server like you see in here. We have lab solution CS which can connect more instrumentation in here. Even like not only chromatography system, we can connect a spectrophotometer system, and even we can connect another brand. So we under one platform software, we call it lab solution. So you can controller, you can reporting, you can do analysis in everywhere you want. As well, it in the same domain, in the same network. Okay. Like you see before, is it the newest digital transformation for your laboratory? Because another product, even like Lab Solution CS, we have a LIMS, laboratory instrument management system. We have QMS, we have DMS, and Nickel training. So is it a full package for your digital transformation of your laboratory? Okay, this is the digital technology has been growing rapidly. So maybe we know we cannot notice if we need the technology as like analytical intelligence for our activity. Okay,
Excuse me, Mr. Alfian, you will have 3 minutes left. Thank you.
Okay, this is actually the last slide from
Okay, that's great.
Okay, so because we don't have much time in here, please, I want to make sure I want to point to you if you have another inquiry. Is it like not only you need an instrument, if you need the method or you need to maintenance or service or something like that, please don't hesitate to call me, to call my team, to call my company, to info our customer service in here. You can save my contact number in here, and you can reach my email in here in alfian@detectjaya.co.id or sales@detectjaya.co.id, and you can visit to our website. Is it detectjaya.co.id. Please don't hesitate to contact us in here. Okay, thank you, and see you, and nice to see you all in the great webinar today. Thank you. I will back to our moderator.
Thank you, Mr. Alfian Mauludin, for a great presentation. Ladies and gentlemen, before continuing to the next agenda, there would be some documentary videos from our partnership that we can watch. So let us watch the video together. Here we go.
Since 2019, PT Wadia Prima Mulia has continued to develop in meeting the needs of basic laboratories for Indonesian territories. Until now, our company has been trusted as an authorized distributor for 11 companies that produce the best laboratory equipment in the world. Our highly skilled and experienced sales team spread across Indonesia from Medan, Jakarta, Bandung, Semarang, and Surabaya. Our products. Sales services, including application and technical support, installation and training, also service and maintenance.
In
the life sciences, researchers are actively pursuing studies aimed at understanding life, the causes of disease, and achieving breakthroughs in regenerative medicine. To observe and accurately measure intracellular processes and structures, researchers need a microscope that combines sensitivity and speed. Increasingly, scientists require the capability to view cells and tissue at high resolution, as well as their larger context with macro to micro capability. All of these advanced features must be easy to use so that even novice users can acquire high-quality data quickly and simply. Olympus created the FV3000 series to satisfy these needs. This next generation of confocal laser scanning microscopes combines leading-edge imaging features with renowned Olympus optics.
All right, the next video. The next video for the host. While waiting for the host, okay, here's the video.
Halo, kami dari PT ITS Science Indonesia. Hari ini kami akan memberikan penjelasan singkat mengenai pengujian porcine. Mengapa perlu melakukan pengujian porcine pada produk pharmaceutical? Beberapa macam bahan baku atau obat, termasuk tablet, kapsul, injeksi, dan sebagainya, dapat berasal dari produk hewani atau turunannya. Contohnya seperti gelatin, yang sering dipakai sebagai bahan utama kapsul atau stabilizer pada vaksin. Selain itu, antikoagulan seperti heparin juga dapat berasal dari porcine. Sedangkan berdasarkan Undang-Undang Nomor 33 Tahun 2014 tentang jaminan produk halal, produk yang masuk, beredar, dan diperdagangkan di wilayah Indonesia wajib bersertifikat halal. Oke, jadi bahan apa saja yang dapat mengandung porcine? Kebanyakan senyawa tersebut merupakan antikoagulan, digestive supplement seperti enzim lipase, amilase, insulin, dan sebagainya. Wah, cukup banyak ya. Tapi tenang saja, pengujian porsain ini tidak sulit kok. Ada beberapa metode yang dapat digunakan untuk mengecek kandungan porsain pada produk. Yang pertama, lateral flow device atau test pack. Metode ini cepat dan mudah, tanpa perlu banyak peralatan laboratorium yang canggih, tetapi sensitivitasnya rendah. Metode 2, ELISA. Waktu preparasinya dibutuhkan 2-3 jam. Preparasinya cukup mudah dan dapat menguji dalam jumlah banyak dalam satu kali running. Dibandingkan lateral flow device, ELISA lebih spesifik, tetapi masih memiliki batas deteksi sensitivitas yang sedang. Metode 3, real-time PCR. Metode ini adalah golden standard untuk pengujian porsain pada produk pharmaceutical dengan limit deteksi yang sangat rendah 1-5 kopi DNA. Pengujian porsain pada produk pharmaceutical sebaiknya dilakukan dengan metode PCR karena bahan baku yang diuji telah melalui proses pemurnian atau pemanasan tinggi sehingga kemungkinan yang tersisa hanya materi genetiknya saja. Selanjutnya, tahap real-time PCR ada 3 yaitu preparasi sampel, ekstraksi DNA, dan amplifikasi dengan menggunakan mesin real-time PCR. Untuk lebih jelasnya, mari melihat alat real-time PCR dari Roche, Light Cycler 96. Alat berkapasitas 96 well ini memiliki 4 channel fluorescence yang dapat digunakan sekaligus dalam satu kali pembacaan. Sebelum memulai proses PCR, profil temperatur dan protokol PCR dimasukkan ke dalam mesin. Setelah itu, tekan tombol start. Secara perlahan akan terbentuk grafik sinyal seperti ini. Sinyal ini terbentuk dari proses denaturasi, annealing, dan elongasi yang berulang, sehingga fluorofor yang terkandung di dalam master mix dapat berpendat dan tertangkap oleh sensor mesin. Sinyal yang didapat kemudian dianalisis menggunakan software Light Cycler, sehingga terlihat sampel mana yang positif atau negatif. Demikian penjelasan singkat mengenai pengujian porsain dari kami. Sekian dan terima kasih.
Now we will move on to the next video.
Halo, kali ini saya mau memperkenalkan alat laboratorium dari Syngene. Ingenious Tree. Sebelum pengenalan alat, apakah Anda pernah mengalami kesulitan saat mendokumentasikan hasil gel electrophoresis atau plot Anda di ruang gelap? Sistem dokumentasi gel secara manual terdiri dari ruang gelap, kamera, dan transluminator sederhana. Dokumentasi gel secara manual seperti ini kadang menyulitkan dan belum lagi kalau hasil foto gel yang didapat ternyata blur. Dengan Ingenious Tree, Anda dapat memilih pencahayaan sinar UV, sinar biru, atau putih, sesuai dengan pewarna yang Anda gunakan. Selain itu, software Genesis akan membantu Anda memilih pengaturan cahaya dan filter yang tepat, sehingga sangat memudahkan untuk mendokumentasikan gel fluorescence ethidium bromida atau cybersafe DNA, atau protein gel dengan composite blue atau silver staining. Untuk penggunaan mode otomatis, Anda hanya perlu memilih tipe gel yang sesuai, kemudian software akan secara otomatis mengatur aperture, zoom, dan fokus serta posisi gel yang sesuai sehingga gambar gel terekam secara optimum. Software Genesis dilengkapi dengan database, tipe gel, dan pewarna yang tersedia di pasaran. Jadi jangan khawatir tidak bisa menemukan pengaturan yang sesuai. Sangat mudah. Hubungi tim ITS Science Indonesia untuk info lebih lanjut. Terima kasih. Stay safe.
And the last video.
Microtomy requires my full attention and dedication to make sure that I deliver the perfect slides. And with the Tissue Tech Auto section, I started getting the job done easier, faster, and with better quality. The automated block alignment makes sure precious tissue is preserved optimally. It is also a great help for making recuts, while the perfect alignment helps me produce even more slides. Trimming and sectioning are done with high precision, each section the same exact thickness. Thanks to the easy operation of the auto section, every technician in our laboratory is quickly up and running. Whatever the need, we are all confident to deliver the perfect slide each time. My name is Mark Shanti, and I'm empowered by Sakura.
All right, ladies and gentlemen, for the next, we will hear a presentation from the Director of PropLife Asia, Mrs. Soni Tababa. Mrs. Soni Tababa, the next 30 minutes is yours. You may proceed.
Thank you very much, Ibu Wiwi. Let me first share my screen and see if, are you able to see my screen now?
Yes, we are able to see it clearly.
Okay, let me just put that into slide mode. And can you hear me well?
Yes.
All right, thank you very much. So I'm standing with, not really standing, I'm sitting right now, but I'm just the last speaker and your break is coming soon. But first of all, I'd like to thank the organizers of ISIBIO 8. This is a lot of work. Congratulations to BRIN and to KBI putting together all this conference, all the researchers with all this fantastic work they have done. And annually at that, since 2012, is really a lot of work. This is, CropLife Asia has a similar, I manage a similar program for 15 years annually, and it's a lot of work. Very much big congratulations. And the other one I'd like to say is that I just would like to thank you for our organizers and participants for allowing us, giving us a space to share our thoughts around plant science, around innovations, as well as around food security. And in the next few slides, before I go to the next few slides, so I am Ibu Soni, and right now I am in Philippines, in Los Baños, Laguna, but I work as a Biotechnology Affairs Director in CropLife Asia, which is based in Singapore. So the pandemic brought me another blessing to be with my family, spending more time with my family while working from home. So let me go next to my next topic, which is just to give you a flavor about CropLife Asia and its network. I always would like to introduce first CropLife Asia. We are a regional organization, and our global organization is CropLife International. CropLife Asia is based in Singapore. We have 15 national associations all the way from North Asia to Southeast Asia and down under. And you can see on your screen the 6 global R&D companies that are our members, and they are very much active in crop protection, seeds, and plant biotechnology space. And I said we have 50 national associations. You see them on screen, and I'm very happy that in Indonesia, we have CropLife Indonesia, which have been in Indonesia helping and contributing to societal goals in Indonesia is CropLife Indonesia, more than 3 decades there. And for our biotech program, you would notice that some of our national association focus mostly on crop protection. So for biotech and seeds, we partner with other organizations like in India, the Federation of Seed Industry of India, where they have a special interest group, which is Alliance for Agri-Innovation in India. We have the Council of Biotechnology Information in Japan, a CropLife international affiliate, and also China is so big that we have to kind of separate the program from a crop protection, purely crop protection, and for biotech. And as you see, we have 15 national associations here. Most of CropLife bring together the advocacies on not only crop protection, but plant biotech and seeds, but others just focus on crop protection. And CropLife associations, whether they are in the region or whether it is CropLife Indonesia, our vision is really to enable a productive food and agricultural systems through innovative crop science and technology. The vision is to have a living standards and improve living standards for not only our farmers, but for those who are using, who are involved in farming in a very sustainable way, and ultimately to improve and contribute to the important goal of food security. And so you would notice that our company members are really into developing innovations in crop science. So the major stakeholders where these technologies will be accessed are by our farmers. So we are hoping that our farmers will be able to grow more on less area through these innovative technologies, so that later on you could see that growing more in less area would help in our advocacies and our goals, overall societal goals in biodiversity conservation, and even in reducing the impact of climate change or in mitigating climate change. So just as a background here, CropLife, whether we talk of CropLife Indonesia, we have programs around crop protection, plant biotechnology, seeds, public affairs. Stewardship is very important. Stewardship is responsible care and anti-counterfeiting. And, you know, when it's a good product, it is likely to be copied. So we have to be very careful as well and make advocacy around anti-counterfeiting so that our farmers would have the guarantee that the products that they are using are genuine and would be able to benefit from it. So I'll just focus my next talk on global events in 2021. I think what I can say about 2021, there are so many big events that impact the way policy will be crafted in the next few, maybe in the short term, 3 years. But this will also impact us being scientists, or even from the industry. So let me just talk a little bit about COP15. So COP is a big police, but it stands for Conference of Parties. And this is about the Convention on Biological Diversity. So it is supposed to have been held in 2020 because this is held every 2 years. But because of the pandemic, this was started to be held in October this year. A high-level segment was started in China, in Kunming, and the theme is ecological civilization. So as you know, this is Convention on Biological Diversity, but this Convention on Biological Diversity is about conservation, about the use of biological diversity, and also benefit sharing. And so we have subsidiary agreements. So you must have heard about the Nagoya Protocol on the Cartagena Protocol first on biosafety, which is the only legal agreement around living modified organisms. You have the Nagoya Protocol on access and benefit sharing, and you have the Nagoya Kuala Lumpur Supplementary Protocol on liability and redress. So as you would note here in Kunming this October, last month, the high level, the parties to this called for urgent and integrated action to reflect biodiversity conservation in all sectors of the global economy. So whether you are in trade, so everybody is enjoined to really be accountable for biodiversity. So it's not limited to a few actors. And there's a call for increased investment and more private sector involvement. So there's a website here. This is where you can get updated news around what happened to COP15. This is a continuing, there's an ongoing negotiation that will be held in person next year in January in Montreal. So probably there will be some coming from Indonesia as party to the Cartagena Protocol to negotiate in this conference, in this convention. The other one I'd like to call is the UN Food System Summit. This one really focused on our food system so that we are called to deliver not only on the 17 Sustainable Development Goals, but really asking member states, the parties to this, to account, to make a reporting. And so, and make more solid commitments towards more sustainable, equitable food systems. So before the summit that was held in September, there were many independent summits and also summit by member states. In fact, as early as January, Indonesia held that summit around the Food System Summit. And so that is very important because maybe in the next 3 months or in beginning 2022, we will hear our leadership in different ministries to talk and set directions around our food systems, around our biodiversity. And the last one I will mention is around the Climate Change Conference, which recently happened in UK, in Glasgow. So this one really, they put together solid commitments. All the parties, they were asked to make their commitments instead of 2040 2040, move it earlier so that you can have global net zero emission. And the other point of this climate change conference is they also mention agriculture as making it more resilient to climate change. And if I just want to bring to your attention, just in case it has missed into your radar, that last month, the ASEAN adopted the framework for circular economy. And so here I put together the strategic priorities. It's around harmonization and mutual recognition. And we are talking now of circular products and services, around trade, around innovation, finance, so energy. And so these are, it encompasses a large sphere of the things that we are doing, and it will impact also the way research will be done and the directions of research that will be done, whether you are in the public sector or in the private sector. And we heard Vietnam is already, the Minister of Agriculture in Vietnam is already very interested in discussion about circular agriculture, and Thailand is also very interested. I have yet to hear from Indonesia how is the discussion around circular agriculture in particular. But it's very interesting. This is something that, everybody must be engaged. And so if we talk around, we go back, we remember Convention on Biological Diversity, Food System Summit, the climate change, this circular economy. I'll just highlight this common thread, not just the three of them, but for this talk, I'll just highlight the commonalities among those developments around the important role of agriculture, of innovations, technologies, and of partnerships. And this is so later on, I'll talk about that, but let me just segue a little bit and go back to the farmers, because whether it's climate change or whether we look at the food, every time we eat, we should thank the farmers. And because they are the frontliners, they are our heroes, they produce our food, but they are facing a lot of challenges, whether it be from climate, because climate could bring extreme weather, more rains or more severe drought, or more pests or changes in weeds and more diseases. And so these are a lot of challenges that farmers have to face, and we are also asking him, hey, produce more, produce sustainably. And so there's a lot of responsibilities that we bring on to the farmers, but we should also be in a position to be able to help the farmers, and that's why we're having this conference. So a lot of the disciplines that will be presented here will be able to find their way to farmers' hands. I'll just go back. This may be a 2014 research from the International Food Policy Research Institute, but this just highlights how important the contribution of technology in Indonesia. So whether they be in crop protection, they can increase yield as high as 20%, or in having crops that make use more efficiently of nitrogen, or having drought tolerant or salt tolerant crops. So when there is extreme weather as a result of this climate change, we see more the technology as a very important intervention to help mitigate or adapt in climate change. So this is another example, no-till. So for example, no-till help in not only in carbon sequestration, but it also double the maize. So if you don't till, you help in carbon sequestration, but it also help in improving yield. And this is the data, again, coming from the IFRI, which is a think tank research agency under the consultative group of international agricultural research. Now, just zeroing on some of the technologies in supplying food sustainably sustainably, particularly from a plant breeder's perspective, they can do a lot. They can develop varieties that can protect or reduce insect damage. They can develop novel herbicide tolerances or disease-resistant varieties, as mentioned with Master earlier, improvement of varieties that are more resistant to pests and diseases. And then we can also develop varieties that can, or farming systems that can improve carbon sequestration or improve tolerance to salinity, drought, or even improve the nutritional profile. And with the advances in science, we see, and we have been hearing, you are all scientists and researchers, so you have heard how novel molecular techniques and transgenics help in that direction. So Master earlier said that I'm very happy to hear that GM potato, earlier there was GM sugarcane, and now GM potato is now advancing, and maybe very soon we can see it really in the market after the variety registration approval of GM potato, and that is very good. And now we also heard from Master the gene editing, and so those are very important innovations. Using these techniques to improve the varieties is a very important way of helping farmers cope with their challenges. I am in my last slide, but this is where I would like to, I would say this is a distillation of my so many years already. I started working for government in 18 years with the Department of Science and Technology in the Philippines in R&D management, and then move on to a regional agency for agriculture under the ISIBIO, in communicating plant biotechnology. And this slides seem for me like a distillation of, also I share the same thought of what are the elements in the environment that could enable plant science innovation. Not only enable, but you would have timely access. I think timely and access is very important to that. So you would see here a lot of bullets, and I just would like to say that these are not exclusive. They impact on one another: political will, science and evidence-based regulation, societal license. So for example, when I worked in the Department of Science and Technology in the Philippines before, then when we tried to craft the crop biotechnology research focusing on our Philippine export winners, and then rice and corn, we have to reach out to our policymakers, not only to our heads of agencies, but even the legislative. We have to make sure that regulations are there, and also risk-based and science-based. And just one thought about regulations, from a biosafety point of view, we have regulations. So for example, Indonesia, they have the regulation as early as 2005. And so over those years, Indonesia has gained a lot of regulatory experience. And with that regulatory experience, you can get to a more comfortable and confident level how to make it more efficient. Societal license is very important. If consumers, so like the GMOs, there's until now, you can still hear a lot of people against it. You can still hear a lot of misinformation. So, and that is making our politicians nervous as well. And that is also making our regulators, slow down or just kind of take a little bit time, because, when there's a lot of noise, that is also affecting the decision making. But here we really have to also listen to our farmers, because farmers need choices. And you are researchers, and not only researchers, when you do into commercializing the products of research, we have to increase our investment. Public investment to research must be sustained and must be increased. IP protection, intellectual property. This is very important because when you invest in research, you also want to gain something from your investment. And the last one here is public-private partnership. Whether it's a global event or global agreements, public-private partnership is important because nobody can exist as an island. And so we're hoping that CropLife Indonesia has been, all these elements, they've been working on this, engaging our governments, engaging the different stakeholders, that someday we can add more choices to the crops that are available for farmers, not just GM sugarcane, not just GM potato, but also GM corn and the other, I heard from Pat Master, the GM tomato and all the other products in research still on gene editing. We really hope that those can come on the ground commercially, that it can be commercialized, be at the hands of our farmers, and inevitably that will redound to the benefits of consumers. So that will be my last parting thought. And with all your help, we can all live in a better planet. Thank you so much for giving us this opportunity.
Thank you, Mrs. Sonita Baba, for the great presentation. Ladies and gentlemen, now we will have a break for an hour and we will start the parallel session at 1:30 p.m. Jakarta time. Thank you.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development, and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by various research institutes, and have more than 70 research centers and supporting units. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Halo sahabat LIPI, selamat datang di video tour Biosafety Level 2 dan Biosafety Level 3 LIPI. Lembaga Ilmu Pengetahuan Indonesia atau LIPI merupakan lembaga penelitian yang memiliki fasilitas Laboratorium Biosafety Level 2 atau BSL2 dan Biosafety Level 3 atau BSL3 yang terletak di kawasan Cibinong Science Center, Bogor. Laboratorium ini digunakan untuk melaksanakan kegiatan penelitian yang melibatkan organisme dan patogen yang berbahaya. Saat ini, kedua laboratorium tersebut telah ditetapkan sebagai laboratorium pemeriksa COVID-19. Biosafety Level, atau biasa disingkat BSL, adalah suatu tingkat sistem keamanan yang dibuat untuk menangani bahan biologi berbahaya di laboratorium. Tujuannya untuk melindungi personil yang bekerja di dalam laboratorium, juga lingkungan dan komunitas sekitar dari paparan mikroba patogen yang sedang diteliti atau diuji. BSL memiliki 4 tingkatan, dari 1 sampai 4, di mana semakin tinggi tingkatannya, maka semakin khusus fasilitasnya, dan semakin berbahaya bahan biologi yang dapat ditangani di fasilitas tersebut. Perbedaan utama dari tingkatan ini adalah sistem tata udara, tekanan, kelengkapan fasilitas, dan alat penunjang lainnya. Kita ke laboratorium BSL-2 dulu yuk! Di dalam BSL-2 ini, ada banyak ruangan yang digunakan sesuai fungsinya untuk deteksi virus SARS-CoV-2 sebagai penyebab pandemi COVID-19. Ruangannya terdiri dari 1. Ruang penerimaan spesimen 2. Ruang logistik sebagai tempat penyimpanan bahan kimia dan bahan habis pakai 3. Ruang ganti untuk pemakaian alat pelindung diri 4. Lab ekstraksi yang terdiri atas ruang antara, ruang laboratorium, ruang mandi, dan ruang dekontaminasi limbah 5. Lab PCR yang terdiri atas ruang antara, ruang pre-PCR, ruang post-PCR, dan ruang analisis. Nah, kalau tadi sahabat LIPI sudah ke BSL-2, yuk sekarang kita ke BSL-3. Fasilitas laboratorium BSL-3 LIPI memiliki infrastruktur yang lengkap dan telah tersertifikasi sesuai standar WHO sejak tahun 2018 dan terus diperbaharui setiap tahun. Biosafety Level 3 memiliki 4 laboratorium, yaitu Clean Rooms, yang terdiri dari Health Biotechnology Laboratory, Food Biotechnology Laboratory, Biosafety Lab, terdiri dari BSL-3 Laboratory, Animal BSL-3 Laboratory. BSL-3 LIPI terdiri atas 2 ruangan. 1. Ruangan mikroba. Ruangan ini digunakan memperbanyak atau mengisolasi materi genetik bakteri untuk digunakan dalam kegiatan penelitian. Misalnya, pencarian obat baru, vaksin, dan sebagainya. 2. Ruangan kultur sel. Ruangan ini digunakan untuk mengkultivasi virus di dalam sel mamalia hidup. Sedangkan Animal BSL-3 terdiri atas 2 ruangan. 1. Ruangan isocages. Ruangan ini digunakan untuk memelihara hewan pengerat yang digunakan sebagai model pada uji di laboratorium. 2. Ruangan pembedahan. Ruangan ini digunakan untuk melakukan pembedahan hewan dan mengambil sampel organ tubuh dari hewan. Nah, untuk mendukung pemerintah dalam mempercepat penanganan COVID-19 di masa pandemi, ruangan di Animal BSL-3 ini dialihfungsikan untuk mendeteksi COVID-19. Sehingga, semua ruangan di Lab BSL-3 digunakan secara aktif untuk semua kegiatan deteksi dan riset SARS-CoV-2. Seluruh ruangan di BSL-2 dan BSL-3 dilengkapi dengan berbagai peralatan yang mendukung aktivitas penelitian, juga didukung oleh sumber daya manusia yang terdiri atas para peneliti dan praekayasa terlatih, tim logistik, dan tim administrasi. Melalui fasilitas laboratorium BSL-2 dan BSL-3 yang dimiliki, LIPI siap berkontribusi secara maksimal untuk mendukung pemerintah dalam mempercepat penanganan COVID-19. Sahabat LIPI dimanapun berada, jangan lengah, jangan menyerah, dan jangan lupa untuk selalu mematuhi protokol kesehatan dimanapun berada ya. Bersama, kita bisa melawan pandemi ini. Salam sehat dan semangat ya!
Ladies and gentlemen, welcome back to ISIBIO 2021. In the following hour, we will have the first parallel session that are divided into 3 breakout rooms. Room 1 and Room 2 will begin with the invited speakers. Room 1 with Professor Anuraga Jayanegara with the topic Modification of Polyunsaturated Fatty Acid Metabolism in the rumen and their deposition in animal products by dietary polyphenol. At Room 2, we'll have Dr. Yopi with the topic Center of Excellence for Integrated Biorefinery as a platform for developing Indonesia's biotechnology research in the future. And at Room 3, the session with the invited speaker will start at 2:30 p.m. Jakarta time with Professor Nicole J. De Vogt with the topic Sponge as Essential Elements in Tropical Reef Ecosystem. Parallel Session 2 will start at 2:50 p.m. and the parallel session 3 at 5 p.m. Jakarta time. And all the presentation of parallel session are scheduled according to the latest rundown shared by the committee. And to enter the room, choose the square icon, breakout room, on the toolbar below and click join next to the room that you choose. Now you might enter the breakout rooms. Thank you.
Mbak Wiwi. Selamat siang Bapak Ibu sekalian.
Selamat siang, Dr. Yopi.
Bu Hermit, selamat siang Bu.
Selamat siang, Bapak.
Ketemu di sini ya Bu ya?
Iya, Bapak.
Good afternoon, Prof. Hermin and Dr. Yopi.
Good afternoon.
Thank you very much for your time. Welcome to Breakout Room 2, and to all participants and presenters, welcome to Breakout Room 2. We are going to have invited speaker and parallel session 1 of Room 2. For these sessions, we'll be guided by Professor Hermin Pancasakti. Professor Hermin is a professor at Biology Department, Faculty of Science and Mathematics, Diponegoro University. She has a lot of experience in teaching, as well as other roles as reviewers and assessors. So without further ado, to Professor Hermin, the screen is yours.
Thank you so much, Ms. Eva Agustriana. Ladies and gentlemen, first, I'd like to greet all participants joining this event. Dear honorable presenters, Dr. Yopi and fellow participants, Assalamualaikum warahmatullahi wabarakatuh. And good afternoon. In this session, our invited speaker is Dr. Yopi, Head of Center for Research and Human Resources Development from National Standardization Agency of Indonesia, or BSN. And Dr. Yopi will be discussing about Center of Excellence for Integrated Biorefinery as a platform for developing Indonesia biotechnology research in the future. Before we begin, allow me to give a brief CV about Dr. Yopi. Dr. Yopi started his career as lecturer at Chemical Engineering Department, Indonesia University. On 2010 until 2015, he was head of Bioprocess Division, Research Center for Biotechnology, LIPI. From 2010 until 2019, Dr. Yopi was head of Biocatalyst and Fermentation Laboratory, Research Center for Biotechnology, LIPI. On 2019 until now, Dr. Yopi was head of Center for Research and Human Resources Development. The specialty and research experience of Dr. Yopi was on bioprocess engineering, biotechnology, standardization, integrated biorefinery, biofuel, and biochemical product, and also bioremediation. And Dr. Yopi was achieving some awards and research grants from Indonesian government and outside the country, like Ristek-Dikti, DIPA, GST-JICA Satelit Project, Indonesian Torres Science Foundation, and also Mombasa Scholarship. Dr. Yopi also have a lot of publication in reputable international journals. For examples, on Applied Microbiology and Biotechnology Express, on Bioresources Technology, and Biotechnology for Biofuels, and International Journal on Advanced Science, Engineering, and Information Technology, and also in International Journal of Systematic and Evolutionary Microbiology. Dr. Yopi also have a book about oligosaccharides, and with the journal of Dr. Yopi have the topic mainly about Manan, catalyst, and bacteria. I think this was a short CV about Dr. Yopi, and now, ladies and gentlemen, please join me in welcoming Dr. Yopi and deliver his presentation. Dr. Yopi, the time is yours.
Terima kasih, Bu. Saya izin share dulu ya, Bu ya. Betul.
Oke.
Ini ya, Bu ya. Sudah terlihat ya, Ibu Hermin ya? Oke. Baik, terima kasih. Thanks Ibu Hermin for introductions. Assalamualaikum warahmatullahi wabarakatuh. First, I would like to thanks to the organizing committee for inviting me today to be a speaker on these sessions. I'm very happy to be here, and thanks to all participants today, because this is the first time for me as a speaker in this seminar. So this time allow me to deliver my presentation related to the Center of Excellence for Integrated Biorefinery as a platform for developing Indonesian biotechnology research in the futures. The content of my presentations today was obtained from the experience as a researcher at LIPI, especially in the Research Center for Biotechnology, start from 2005 until 2018, before I'm joining to the National Standardization Agency of Indonesia. So today focus my presentation related to the integrated biorefinery, not about the standardizations. So next slide. Okay. This is the outline of my presentation today. I divide into the 5 groups. First, I would like to explain about the concept of classification of the integrated biorefinery, why we need to establish this research in Indonesia. Second one is strategy to developing integrated biorefinery research group in Indonesia. And the third one, information of output of the integrated research so far, with a focus on the biocatalyzed productions. And the fourth is to establishment the Integrated Biorefinery Center of Excellence. And the last one is the implementation and future planning of this integrated biorefinery. So let's move to the next slide. I would like to explain about the concept of the integrated biorefinery. As you can check the informations and check the journal and reports and website related to this concept of the integrated biorefinery. There's so many information. So today I would like to take one of the interesting definitions which states that the biorefinery system is described as conversion pathway from feedstocks to products via platform and processes. The platform are intermediates from which final product are derived. So this very interesting because there are so many different types of biorefinery in these words. So in this slide, you can see the major difference biorefinery types. There's 9 groups. First is the biorefinery types, which is lignocellulose feedstock biorefinery, mainly use nature-dried raw materials as cellulose containing biomass and waste. And next, the whole crop biorefinery use all whole raw materials. The third one is the green biorefinery, use the nature wet biomass as a green grass, clever and mature cereals. And the fourth is the two platform concept biorefinery, including the sugar and syngas platform. And the fifth is the conventional biorefinery based on the existing industry, such as sugar and starch industry. And they are the thermochemical biorefinery based on the mixed several technology. And also they are marine biorefinery based on the marine biomass, the liquid phase catalytic processing biorefinery, and the last one is the forest-based biorefinery based on the full integrations of biomass for production like pulp, fibers, chemical, and energy. So based on that differences of the biorefinery system in the words, some of the researchers already classifies that the biorefinery system. So this slide show you the classification of the biorefinery systems. So you can see the example on how biorefinery system can be classified. These classifications are broad and very generic, maybe, and provide a little information on the specific characteristics. So classification criteria on this table show is not homogeneous. Some of them refer to the type of the feedstock, like the whole crop biorefinery and lignocellulose biorefinery and green biorefinery. And the others, while others focus on the technology involved, such conventionals and thermochemicals and the two platform concept biorefinery. So there are so many types of the biorefinery system in the world. So next, I would like to show our strategy to establish this integrated biorefinery in Indonesia. First, as a background, we know that the oil refinery has been developed and established well by growing the petroleum industry. And as an impact, as we know that the greenhouse problem is becoming the serious environmental problem in the worldwide level. So it's very important to resolving these greenhouse problems. That is instead of the use of the fossil oil is biomass utilizations as an alternative feedstock for construction of sustainable and low carbon society without fossil oils. So this is our first strategy to utilization biomass with biorefinery technology. It means we need to construct the biomass complex strategy for fuel and chemical production in Indonesia. Next is this is the why we need to develop the integrated biorefinery in Indonesia, as we know that the fossil oil stock is decreasing right now in Indonesia, and on the other hand, the energy supply and demand are increasing. This is depend on the population's growth in Indonesia. To challenge this condition, Indonesian government already released the regulation related to the strategic of target the use of the energy mix. So government try to reduce utilization of the fossil oil 20% until 2025 and support the increased utilizations of the biofuels, including the bioethanol and biodiesel 5% until 2025. So this is the important point related to the government's decisions. Next, I would like to show that there are two important points.
Recording in progress.
Basic capital of the this integrated biorefinery is important developed in Indonesia. The first one is the ability of the biomass in Indonesia compared to the start-based non-start biomass, lignocellulose, is very promising, interesting. As a biomass feedstock, the cellulosic material are better for utilization because no competition problems between foods and non-edibles. So, and there are very abundant this lignocellulose biomass in Indonesia from agriculture, horticulture, industrial waste fractions. So cellulose-based biomass can be degraded by microbe, of course, and convert into the valuable a lot of products such as biofuel and biochemicals. But the problem is, what's the right technology to convert, to hydrolyze the very big abundant biomass and convert into valuable products? So we need the efficiency process. That's why the biotechnology approach becomes very interesting to do, especially for the development of special microbes and their derived products such as biocatalysts, which are important for degrading the whole raw biomass, cellular biomass, to convert into the valuable products. This is this slide show that after we have the abundant biomass in Indonesia, we have a huge number of the culture collect of Indonesia. LIPI, previously, now we call BRIN, already established the Indonesian culture collections. This is the International Standardized Microbial Resource Center, first one in Indonesia and only one, and already have collection more than 10,000 strains, including bacteria, actino, fungi, and others. So we can use this kind of culture collection to be explore more. We have to do research together, collaboration work with a lot of researchers from Research Center of Biology, Biotechnology, Biomaterial, to get good microbes from these culture collections. So the key point is how to use this collection of microbes to support the research of integrated biorefinery in Indonesia. Okay, next, I would like show you a little information about oil refinery, the paradigm shift of oil refinery into biorefinery strategy based on the biotechnological approach. Okay, the main key technology that needs to be developed is to create the microbial cell factory. This one, okay. So with the genetic engineering approach for the degradation process, of course, the various existing cellulose biomass in Indonesia to produce the target products such as biogas, biofuel, and biochemical products. So this is our strategy to do in the integrated biorefinery in Indonesia. Next, this is I would like to show the integrated biorefinery works process. The initial stage of the start from the treatment, the target biomass. Okay, we need a physical chemical treatment in this field process. Then the following step is to degrade the biomass into the biomass polymer into the small monomer. We call it C2 and 6-3 building blocks productions using the biocatalyst or the engineering bacteria. Then the conversion process of this building block into the first intermediate products such as bioethanol, lactic acid, amino acid, and others. So in this point, biotechnology and bioprocess approach become the main technologies. Okay. In the next step, if you want to variations of the products, other derivative products, it is necessary to apply the chemical technology and maybe the physical technology, such as the production for polylactic acid from lactic acid. We know that polylactic acid is a bioplastic right now. So there are a lot of derivative product from this building block. It depends on the chemical process methodology you choose. Okay, So from this slide, we know that it's shown that the implementation process of integrated biorefinery strategy require a lot of collaboration of multidisciplinary researcher and scientists. Okay.
Excuse me, Dr. Yopi, your time is almost end.
Okay.
Thank you.
Next, I would like to show several strategy to developing integrated biorefinery research. Actually, in Indonesia, there are a lot of researchers already conduct a lot of research related to bioprocess and biorefinery. This is our colleagues from BPT, LIPI, of course from ESDM and others, and all of these member scientists already linked in the Indonesian Consortium of Biotechnology and Asian Federations. And Indonesia, and this is the prospect related bioprocess and biorefinery research in LIPI. So we already have a lot of experience to do research related to the microbes aspect, bioprospecting, exploring the biomass, of course, upgrading the technologies related to biomolecular, bioprocess engineering, and supporting the quality source of the researcher, human research, and also facility. Okay. So, but we do know that the weakness of this kind activity is most of the activity research conducted by Indonesian researchers is partially conducted, not integrated. So that's why we need to special strategy to strengthen and develop the integrated biorefinery research in Indonesia. So before start, we have to check the state of the art of the biorefinery research in the world. So we check that the developed country already set up the program related to the integrated biorefinery, but the very interesting is from Japan. They have the special program, especially in the Kobe University, already established the innovation bioproduct Kobe. So we choose this innovation bioproduct Kobe program as a partnership in our future collaborations, because the main topic research activity is very similar and it's very important for us. So we do several communications, coordination, and together submit the proposal to conduct the collaboration research. This is the first one, collaboration research, funded by GSPS LIPI bilateral project related to the production bioethanol mono-oligosaccharide production using the recombinant yeast cell surface engineering. So in this program, we focus on the research utilization of the palm kernel cake to produce bioethanol and functional food using the consolidated bioprocessing. So we succeeded construct the recombinant yeast displaying Mananasi genes. This is the first report in the world. We already published in the bioresearch journals. You can check, and the result is very good. So based on this experience, we do more research activity to expand the topic. So before that, I would like to explain about how the differences this development of consolidated bioprocessing compared to the previous technology, such as spirit hydrolysis of fermentations on simultaneous saccharification and co-fermentations. Using the consolidated bioprocessing, using the super microbial cell based on the arming yeast, and we can call as a cell surface display technology. All compared to the previous one, the process for the productions like bioethanol, for example, from biomass, we can do in one stage. So using the consolidated bioprocessing, we can reduce the cost, the whole cost, the save energy, and simplify all of the process. So this system integration is very good for the futures technology development in the futures. Okay, so Using that kind of technology, we expand our activity, research topic. Then we develop a partnership with the Kobe University and succeed to get a large fund from GST JICA SATRIPS projects. We in this show you can see that in this project we focus in the waste biomass from palm oil industry to produce bioethanol and bioplastic. We development 5 key technology, pre-treatments, microbreedings, enzyme fermentations, chemical synthesis, and the feasibility studies of the whole process, including the implementation of the consolidated bioprocessing technology. So this is the main structure of this project. We have around more than 40 maybe scientists involved in these projects, from RC Biotechnology, RC Chemistry, RC Biomaterials, Biology, and also Chemical Engineering Department for Indonesia University. We divided the main tasks based on the 5 outputs, and that need to be achieved during 5 years projects. Okay, so as we described before, that the main technology that we have to develop is the cell factory. This slide shows the application of cell factory is integrated biorefinery. We can choose and create a cell factory based on the type of the microbes, which is some from bacteria or from fungi like Aspergillus or Saccharomyces cerevisiae. So we can customize and adoptions this kind of cell factory based on the target product, such as biofuel and bioproducts. Using this cell factory, the process is very efficient and have high novelty and high advanced technologies. So how to build the supermicro cell factory? First, we have to decide the candidate of potential gene. Next is the candidate of potential of host cell. This is from bacteria, actino, or others, because it is very important to make sure that the gene that we clone is suitable for the host cell candidate that we constructed. Okay, this is very important thing, but it's very difficult ones. So we are now is still in ongoing process to making this kind of host cell and the system expressions for the production protein and chemical products. So next is the for related to the results of our activities so far. We already developed several enzymes like the mananase, silanase. We already like developed the screening integrated system, screening protocol from targeted enzyme. Of course, we developed the construct protein expression systems and other things. Not of all of them already fully succeed, but still carry out because there's some constraints, so we need still move on to get a good results. So in the future, we hope can do like cooperates with various stakeholders, research from others research institute to develop this kind of industrial enzyme. Okay, this sample of the results. We do screen a lot of Indonesian culture collections bacteria and get already 6 microbe that highly positive produce targeted enzyme already cloned and check characterize the recombinant ones, and also got the candidate for production system actinomycetes. Okay, this is the slide show that we already have one candidate, the good one, compared to the Streptomyces lepidianus control positive. We have a Streptomyces albus that can produce during only 1 day for the cell productivity. So it's very good for the target candidate for the host cell.
Dr. Yopi,
Are still
maybe you must quick.
Okay. Next is the enzyme product. We have the mananase and the silanase. You can check the product. Next, maybe for the last one, Ibu Hermin. Okay. We finally, after we apply to Ministry of Research, Technology, and Higher Education, we achieve and establish, got certified as Center of Excellence in the field of the integrated biorefinery in 2016. This is a very high achievement in Indonesia. So we do hope can still doing research related to this integrated biorefinery in the future. So we do hope collaborations with a lot of research institutes in Indonesia, because there's still a lot of cargo collection that we have to be explored. Okay, so there are a lot of target enzyme, and still there are a lot of target of biomass. So there's still a lot of options that we can do together. So we do hope with the brains, there some the priority to conduct and continue this kind of activity related to the integrated biorefinery in Indonesia. And finally, I would like to thanks to all of the members of the LIPI, Kemenristek Dikti, JSJIK Kobe, all of the members, especially Professor Bangman and Ogino Sensei, for supporting all this time. Thank you for kind attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much for the very interesting presentation, Dr. Yopi, and for all the knowledge that has been shared. I'm sure that there is a lot of questions that addressed to you from the participant, but due to the limited time, please allow them to communicate further with you later. Is that okay, Dr. Yopi, for you?
Oh, no problem. Anytime.
Okay, thank you so much. And participant, let's give a big applause to Dr. Yopi.
Terima kasih banyak Ibu Hermin, Bapak Ibu sekalian. Mohon maaf ya Bu ya, agak panjang.
It's okay. Okay, and now I will give the time to the MC.
Yes, Prof. We have parallel session 1 with presenter number 14, Aryo Beta Julian Silvero. For IT team, please play the video.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. First of all, let me introduce myself. My name is Aryo Beta Julian Silvero from Research Center for Biotechnology, National Research and Innovation Agency, or BRINCH. On this occasion, I would like to deliver my presentation with entitled Microbial Lipid Production of Oleaginous Yeast Lipomastix stachyi in a CCY604 in the presence of furfural and 5-hydroxymethyl furfural as the inhibitory chemical compounds. The next slide is an outline of my presentation, which consists of introduction, method, result and discussion, and conclusion. Oleaginous microorganisms have been described based on the ability to accumulate between 20 to 70% intracellular lipid, which containing primarily triacylglycerols and have similar properties with vegetable oils. Oleaginous microorganisms may consist of bacteria, yeast, fungi, and microalgae. and have many applications, especially as the intermediate building block for oleochemicals production, such as fuels, soaps, plastics, additives for food and cosmetic industry. Nowadays, petroleum-based chemistry and fuels are limited sources and non-renewable depletion. And due to the depletion of crude oil, environmental issue and an unstable as well as increasing price from year to year, therefore, to tackle these problems, the expansion studies to a broader panel of oleaginous species would therefore useful for development of more robust industrial strain that able to utilize a broader range of nutrients and tolerate higher level of inhibitors. Several oleaginous microorganisms have ability to accumulate TAGs when grown on hydrolyzed and cellulosic material, such as forestry waste and other agricultural residues. These types of oleaginous have useful properties for research and industrial applications, such as utilization of a broader lignocellulosic biomass or tolerant to inhibitors, which are generated during the treatment of lignocellulosic biomass. Among the oleaginous microorganisms, oleaginous yeasts have many advantages. For example, yeasts have shorter fermentation times, higher cell mass production, higher growth rate, and yeasts have ability to assimilate various carbon sources in lower pH. Lipomyces turkey is one such emerging species among the well-known oleaginous yeast that has garnered attention for its exceptional potential for biotechnology applications. The oleaginous yeast used in this study is Lipomyces turkey in ACC Y604 and provided by Indonesian Cultural Collection or in ACC. The microbial lipid productions are mainly consist of two main stages, which are pre-culture, which to initiate the cell growth, and main culture for lipid production stage. The pre-culture stage were conducted using YMPG medium, and the main culture were conducted using limited nitrogen mineral medium or -NMM medium. To investigate the microbial lipid production in the presence of inhibitory compounds, the -NMM medium contained glucose and xylose as a mixture carbon source, which is 50 gram per liter glucose and 50 gram per liter xylose, was supplemented with furfural and 5-HMF, either individually or a mixture of furfural and 5-HMF. The sugar concentrations during and after fermentation were analyzed using HPLC. Meanwhile, the total lipid quantifications was measured by gravimetric analysis following Fold method. The fatty acid methyl esters profile of reduced microbial lipid was analyzed using GCMS instrumentation. The fermentation profiles obtained for this study, including the sugar consumptions indicated by first y-axis and the cell growth indicated by second y-axis from different typical mediums of microbial lipid productions are shown in this figure. Figure A stands for fermentation profile with glucose and xylose carbon sources as the control, B for fermentation profile in the presence of furfural, C for fermentation profile in the presence of 5-SMF, and D for fermentation profile in the presence of a mixture of furfural and 5-HMF. The closed square refers to cell growth. Closed triangle chart represents xylose consumption, and the closed diamond refers to glucose consumption. Based on these figures, the fermentation time varied from 3 to 5 days of all typical mediums. Generally, in 4 typical mediums, this strain required 3 days to fully consume glucose, whereas for xylose, the time course varied from 4 to 5 days, depend on the medium compositions. Figure A, as the cultivation control, shows that this strain has the ability to assimilate a mixture of glucose and xylose almost simultaneously. Whereas figure B shows the fermentation profile of Lipomyces turkey in the presence of furfural. It was shown the slightly inhibition to the glucose assimilation at the first to second days compared to the control mediums. The glucose concentration in the medium containing furfural was exhausted after 3 days, which has similar period with the control mediums. On the other side, the xylose assimilation was significantly obstructed by the presence of furfural, especially at the first to second day's fermentation. The xylose concentration was depleted after 5 days fermentation, which 1 day is lower than control mediums. The fermentation profile in the presence of 5-HMF, figure C, has a similar pattern with the figure B, where the glucose and xylose assimilation were inhibited during first and second day's fermentation. Figure D, glucose assimilation during 2 days after fermentation was seen weak compared to other conditions. Longer fermentation occurred for xylose assimilation, and the existence of 5-HMF in the fermentation lead to an incomplete assimilation of xylose at the late stage of fermentation. Two furanaldehydes, furfural and 5-HMF, were used in the study to represent the over-degradation of pentose and excess sugar, which generated during the pretreatment of lignocellulosic biomass. Inhibitor concentration was selected for this study that represents range often found in hydrolysates. Furfural was found less toxic to stachy, followed by 5-HMF. There is unknown correlation between the hydrophobicity and toxicity of these two aldehydes. The hydrophobicity of aldehydes, however, is not only determinant of its toxicity. This table shows the microbial lipid production in the presence of inhibitory chemical compounds. The presence of inhibitor in the cultivation medium, apart from being inhibited cell growth and reduced the carbon source consumption rate, it also resulted in a decrease in microbial lipid production. The microbial lipid production by this strain were achieved up to 67%, even though in the presence of inhibitor compounds. In general, the biomass production and lipid content were decreased in the presence of inhibitory compounds in the cultivation medium. Although the microbial lipid produced by stachy were dropped in the presence of furfural and 5-HMF, however, the ability of this strain to accumulate more than 60% lipid has demonstrated that this strain could detoxify furfural and 5-HMF. Even though many previous studies have shown that yeast could metabolize furans, 5-HMF, the precise mechanism involved remains unknown in most cases and should be clarified in the future study. Our data indicated that Lipomyces stachy tend to be more sensitive to 5-HMF than to furfural at the represent range found in
Excuse me, Pak Arya, your time is almost end. Please be quick. Thank you.
The firm composition of Lipomyces stachy grown in 4 typical medium are shown in this table. In general, there were no major differences of FAME's profile between the control medium and medium containing inhibitor compounds. The FAMEs were unchanged by the presence of furfural and 5-HMF, which shows that inhibitory compounds did not significantly affect the fatty acid methyl ester biosynthetic pathway. The major fatty acid produced in all cultivation conditions were oleic acid, palmitic acid, stearic acid, palmitoleic acid, and linoleic acid. In addition, the most major fatty acid produced by Lipomyces stachy in this study was oleic acid, where a large amount of oleic acid has beneficial values as the raw material for biofuel production, which can improve oxidative stability and cold flow properties of biodiesel. Furthermore, high oleic acid has been considerable as an input for many renewable polymers, elastomers, lubricant, and other oleochemicals. From the study, we can conclude that the study exhibited that in addition to obstructing growth, the presence of inhibitor compounds corresponding with decreased microbial lipid content. This effect associated with different inhibitors, 5-HMF, often decreases both growth and lipid accumulation more severely than furfural, and it also strain-specific. Options for diminishing this effect include use of hydrolysate with decreased inhibitor concentration, detoxification of hydrolysates, or selection of yeast strain which have naturally resistant to inhibitors. And this study dispenses support for those wishing to chase the third option. Further validation of this effect of relevant inhibitors on biomass and microbial lipid production by a promising yeast candidate will assist in process development. This chart is illustrated of the many application and transformation of fatty acid derived from microbial lipid via certain chemical reaction. For example, biodiesel production, emulsifier, polymers, petrochemicals, and etc. That's, ladies and gentlemen, was what I wanted to share with you. Thank you very much for your kind attention, and thank you very much.
Next presenter, number 15, Hartati.
Good afternoon. Thank you very much to ISIBIO committee for this opportunity. First of all, I want to introduce myself. My name is Hartati. I work at Research Center for Biotechnology, National Research and Innovation Agency. Today, I would like to share the result of my research, which has a title Potential of Lactococcus lactis strain LAC3 isolated from Indonesian traditional fermented buffalo milk or dadi as antioxidant and anti-diabetic agent. This is the outline of my presentation. I will start with the introduction. Dadi is the fermented buffalo milk originating from West Sumatra. Previous study reported that Lactobacillus plantarum is dominant LAB in dadi from Solok, West Sumatra. Meanwhile, from dadi in Bukit Tinggi, the predominant LAB is Lactobacillus, Lactococcus, and Leuconostoc. Dadi beneficial health effect is due to lactic acid bacteria probiotic properties. During the fermentation process, LAB produces several bioactive compounds such as vitamin, enzyme, GABA, bacteriocin, bioactive peptide, conjugated linoleic acid, and exopolysaccharide. Previous report revealed that Lactococcus lactis subspecies lactis isolated from dadi is known to have antioxidant activity. And LAB isolated from fermented sauce from Sumbawa, colmacin, possess antibacterial, anti-diabetic, and antioxidant activities. In Indonesia, diabetes mellitus is a major health issue due to its capability at 8.5% each year, and more than 10 million Indonesian people living with diabetes. Oxidative stress is the main contributor in the pathogenesis of diabetes mellitus. That's why discovery of alternative antioxidant and anti-diabetic agent is really important. The objective of our study is to investigate the potency of secondary metabolite produced by LAC3 isolated from dadi to provide scientific evidence for further development as antioxidant and anti-diabetic sources. And here are the methods in our study. The first is production of secondary metabolite from LAC3, which is cultured in M17 medium. And the second is two-step purification method. First by ammonium sulfide precipitation, and the second by gel filtration chromatography. Protein then characterized by SDS page and proceed with silver staining to detect protein which are separated during SDS page protocol. Next slide shows the protocol of anti-diabetic assay based on inhibition on alpha-glucosidase enzyme. And this is the protocol for antioxidant activity assay using DPPH. The purified sample, as much as 400 microliter, was then analyzed by LCMS at PUSLAB for Polri. The compound was identified by ChemSpider database and interpreted with MassLink software. And here are the results. Figure 1 shows SDS page and silver staining result of the protein fraction. The peptide band were visualized between 10 to 45 kilodalton after silver staining. The potential fraction, fraction number 1 until number 12 and 44-hour metabolite were used in bioprospecting assay as anti-diabetic and antioxidant agent. And this is the result of anti-diabetic activity of potential fraction LAC3 isolated from dadi. The inhibitory activities from this potential fraction is varied between 82 to 95%. And our result is supported by previous report that reveal LAB possess anti-diabetic activity probably by producing exopolysaccharide, which can inhibit alpha-glucosidase enzyme activity. And Lactococcus lactis could produce secondary metabolite which play a vital role in diabetes mellitus. And the antioxidant activity assay show the fraction number 2 to number 12 give antioxidant ranging from 39 to 89% compared with vitamin C as a control. Our result also supported by previous report which reveal that DADI have antioxidant activity beside Lactobacillus plantarum. And based on bioprospecting analysis using anti-diabetic and antioxidant, we decided to choose fraction number 11 for further analysis by LCMS to identify candidate of active compound contained in the fraction. Total of 17 peak was obtained from LCMS analysis. Each peak represents a different compound. However, some peaks in the chromatogram cannot be identified by the ChemSpider database. The possible compound formula, chemical name, as well as mass, weight, monoisotopic weight, and retention area were defined in Table 3. These 12 compounds have a mass ranging from 143 to 4,363 Dalton. Based on the result of LCMS analysis, among those compounds listed in Table 3, there are several compounds known as antioxidant, such as hydrazine carboxamide, and as anti-diabetic agent, for example, metformin-related compound and metformin hydrochloride. From those results, we can conclude that our studies show that secondary metabolite produced by LAC3 isolated from DADI exerted functional activities as antioxidant and anti-diabetic agent. Further step of purification is needed to determine the active compound as well as in vivo assay to examine anti-diabetic and antioxidant activity. In addition, in silico approach is also needed to study the potential interaction between the compound and the receptors related to diabetes mellitus and oxidative stress. Thank you very much for your attention.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Hope all of you always stay healthy. It's an honor for me being able to present our research paper on the 8th International Symposium of Innovative Bioproduction in Indonesia on Biotechnology and Bioengineering 2021. My name is Dini, I'm from Biology Department ITS, and I will be the oral presenter for our research paper with the title Bioinformatics Study of Starch Synthesis Gene Variation in Cassava and its Potential Use. Introduction. Cassava is known as the rice plant contains starch in the root parts of the cassava plant, as you can see on the picture. Starch are made from amylose and amylopectin through starch biosynthetic pathways. The comprehensive study on starch biosynthetic pathways is potentially valuable approaches to increase cassava starch content. Within the genes, there are starch synthesis gene that's involved to make amylose and amylopectin. Therefore, we aim to identify the nucleotide variations, protein structure, and physicochemical characteristics of cassava starch synthesis gene in silico using bioinformatic tools. To reach the aim in this paper, There are three methods. First is identification of starch synthesis gene. To analyze sequences identity within nucleotides and amino acid sequences of cassava, sweet potato, and potato, and we analyze it through MSA using muscle. After alignment, the phylogenetic tree was constructed using the neighbor joining method with a 1000 bootstrap replicate on MegaX. For the amino acid domain characteristic and motif, the amino acid sequences will begin search the conserved domain using Web series search, and then analyze it, the domain characteristic and motif with some references from literature. And the last method is protein subunit character. For the secondary structure, we analyze it with SIMPA 96. For the tertiary structure, with Pireto, and the physicochemical characteristic with Port-Params expansion. Results and discussion. This is the information of starch synthesis gene variation of cassava and other tuberous plants. The other tuberous plants damages is sweet potato and potato. The function, the type of these three tuberous plants. For the SS has 1 until 6 type, and for the GBSS has 1 until 2 type. For function, SS1, 2, 3 are connected with structure production of amylopectin, while SS4 take responsibility of initiation and morphology starch granule, also with GBSS. This is the result of a phylogenetic tree from SS and GBSS of the three tuberous plant with 1000 bootstrap replicate. From the nucleotide and amino acids figure, there are two clades partitioned by the SS and GBSS gene, and the figure looks like similar. Usually, the phylogenetic tree that we see from here, the SS and GBSS make a sister group with the same type, but there are any different in here. For the nucleotide sequences, the epimebetad SS is one more similar with the solanum tobrosum SS2 with the bootstrap 99. But for the amino acids, the solanum tobrosum SS2 makes sister group with monoplus quanta SS2 with the bootstrap 48. The other differences coming from this bootstrap value that I'm squaring it, it's 95 for the nucleotide sequences and 99 for the amino acid sequences. This is the data for the conserved domain of the SS and GBSS gene. Starch synthesis of the three tuberous plant almost all have domains from glucosyltransferase 5 and glycogen A, except for several types of starch synthesis such as solanum tuberosum SS3 and Manihat esculentase SS3 and 4. The glucosyltransferase is folds into two Ross manifold domains. Between the two domains, there is a catalytic center. For the Ross manifold motif is a motif that consists of supersecondary beta-alpha beta, forming two beta-alpha beta-alpha beta domains. From the N-terminal, it consists of a central sheet with 9 strands which are mostly parallel and tied on both sides by 7 helices. The C-terminal domain exhibits 6-stranded parallel sheet folds and 9 helices. The three-dimensional structure of the protein that represents the conserved domain, and this is accessed using the protein databank database. To the left is the three-dimensional structure from the glycogen A, and the right is glucosyltransferase 1. And this is the data for the secondary structure. The data All of the data have the same pattern with highest percentage is random coil, followed by alpha helix, then extended strand. However, there are exceptions. In Manihot Esculenta SS4, alpha helix is the most abundant structure in the secondary structure of starch synthesis. And this is the tertiary structure data. From this data, we can see the sequence identity were ranging from 51% to 82%. And the use template is based on the number of similarity of template amino acid sequences with amino acid sequences taken from NCBI. And the chain is just from A or B chain. And this is the illustration of the tertiary structure of cassava from SS1 until 4 and GBSS that analyzed using period 2. And this is the physicochemical characteristic data. There are 3 physicochemical characters identified: its instability index, aliphatic index, and gravity. For the instability index is to measure protein stability. If above 40, it means the protein is unstable, and if below the 40, it means the protein is stable. For the data before, the stable protein coming from Ipomea batatas GBSS1, Solanum tuberosum SS1, and Solanum tuberosum GBSS, also Manihot Esculenta GBSS. For the aliphatic index, it means to calculate relative volume used for aliphatic side chains. The higher result coming from Solanum tuberosum GBSS, and the lower is from Manihot Esculenta SS3. For the gravy is to determine average hydropathy value for peptide or protein. Hydropathy value can be correlated with structure and function of protein. The higher result coming from Solanum tuberosum GBSS, and the lower result coming from Manihot Esculenta SS3. Conclusion. The starch synthesis gene variation of Manihot Esculenta with Solanum tuberosum and Ipomea batatas was low based on constructed phylogenetic tree. This indicates that the possible function of the gene is similar among other previously studied plants. The information obtained can be further developed by using bioinformatic analysis of starch synthesis gene annotations, whilst the primary design could be applied for gene expression analysis of cassava using quantitative PCR. Furthermore, bioinformatic information of starch synthesis is potential to be applied for functional analysis of cassava starch synthesis genes through transgenesis. This is my presentation. Thank you for the attention.
Thank you so much for all the presenters. We have 3 presenters, Mr. Aryo, Ms. Aihetati, and also Ms. Tini. And now it is time for us for the question. If all of the participants have the question, can type the question in the chat room. Or maybe the other participant want to ask the question directly. We still have 6 minutes before this this meet to be ended. I think maybe all of the presenter was clear, yes, to all of the participants. And I think it's better for us to close this meet, and the time will be delivered to the MC, Miss Eva.
Okay, thank you very much to Professor Herman for your time, and to Dr. Yopi, thank you very much for your for sharing your knowledge. And we are we have done parallel section 1, and before entering parallel section 2, we are going to enjoy video from our partner.
Thank you so much for all the participants.
Thank you very
Assalamualaikum
much.
warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you.
Halo, kami dari PT ITS Science Indonesia. Hari ini kami akan memberikan penjelasan singkat mengenai pengujian porcine. Mengapa perlu melakukan pengujian porcine pada produk pharmaceutical? Beberapa macam bahan baku atau obat, termasuk tablet, kapsul, injeksi, dan sebagainya, dapat berasal dari produk hewani atau turunannya. Contohnya seperti gelatin, yang sering dipakai sebagai bahan utama kapsul atau stabilizer pada vaksin. Selain itu, antikoagulan seperti heparin juga dapat berasal dari porcine. Sedangkan berdasarkan Undang-Undang Nomor 33 Tahun 2014 tentang jaminan produk halal, produk yang masuk, beredar, dan diperdagangkan di wilayah Indonesia wajib bersertifikat halal. Oke, jadi bahan apa saja yang dapat mengandung porcine? Kebanyakan senyawa tersebut merupakan antikoagulan, digestive supplement seperti enzim lipase, amilase, insulin, dan sebagainya. Wah, cukup banyak ya. Tapi tenang saja, pengujian porcine ini tidak sulit kok. Ada beberapa metode yang dapat digunakan untuk mengecek kandungan porcine pada produk. Yang pertama, lateral flow device atau test pack. Metode ini cepat dan mudah tanpa perlu banyak peralatan laboratorium yang canggih, tetapi sensitivitasnya rendah. Metode 2, ELISA. Waktu preparasinya dibutuhkan 2-3 jam. Preparasinya cukup mudah dan dapat menguji dalam jumlah banyak dalam satu kali running. Dibandingkan lateral flow device, ELISA lebih spesifik tetapi masih memiliki batas deteksi sensitivitas yang sedang. Metode 3, real-time PCR. Metode ini adalah golden standard untuk pengujian porcine pada produk pharmaceutical dengan limit deteksi yang sangat rendah 1-5 kopi DNA. Pengujian porcine pada produk pharmaceutical sebaiknya dilakukan dengan metode PCR karena bahan baku yang diuji telah melalui proses pemurnian atau pemanasan tinggi sehingga kemungkinan yang tersisa hanya materi genetiknya saja. Selanjutnya, tahap real-time PCR ada 3, yaitu preparasi sampel, ekstraksi DNA, dan amplifikasi dengan menggunakan mesin real-time PCR. Untuk lebih jelasnya, mari melihat alat real-time PCR dari Roche, Light Cycler 96. Alat berkapasitas 96 well ini memiliki 4 channel fluorescence yang dapat digunakan sekaligus dalam satu kali pembacaan. Sebelum memulai proses PCR, profil temperatur dan protokol PCR dimasukkan ke dalam mesin. Setelah itu, tekan tombol Start. Secara perlahan akan terbentuk grafik sinyal seperti ini. Sinyal ini terbentuk dari proses denaturasi, annealing, dan elongasi yang berulang, sehingga fluorofor yang terkandung di dalam master mix dapat berpendak dan tertangkap oleh sensor mesin. Sinyal yang didapat kemudian dianalisis menggunakan software Light Cycler, sehingga terlihat level mana yang positif atau negatif. Demikian penjelasan singkat mengenai pengujian porcine dari kami. Sekian dan terima kasih.
saya mau memperkenalkan alat laboratorium reaksi engine. Ingenious Tree. Sebelum pengenalan alat, apakah Anda pernah mengalami kesulitan saat mendokumentasikan hasil gel electrophoresis atau plot Anda di ruang gelap? Sistem dokumentasi gel secara manual terdiri dari ruang gelap, kamera, dan transluminator sederhana. Dokumentasi gel secara manual seperti ini kadang menyulitkan dan belum lagi kalau hasil foto gel yang didapat ternyata blur. Dengan Ingenious Tree, Anda dapat memilih pencahayaan sinar UV, sinar biru, atau putih sesuai dengan pewarna yang Anda gunakan. Selain itu, software Genesis akan membantu Anda memilih pengaturan cahaya dan filter yang tepat sehingga sangat memudahkan untuk mendokumentasikan gel fluorescent ethidium bromida atau cybersafe DNA atau protein gel dengan composite glue atau surface staining. Untuk penggunaan mode otomatis, Anda hanya perlu memilih tipe gel yang sesuai, kemudian software akan secara otomatis mengatur aperture, zoom, dan fokus serta posisi gel yang sesuai sehingga gambar gel terekam secara optimum. Software Genesis dilengkapi dengan database tipe gel dan pewarna yang tersedia di pasaran, jadi jangan khawatir tidak bisa menemukan pengaturan yang sesuai. Sangat mudah. Hubungi tim ITS Science Indonesia untuk info lebih lanjut. Terima kasih. Stay safe.
Microtomy requires my full attention and dedication to make sure that I deliver the perfect slides. And with the Tissue Tech Auto section, I started getting the job done easier, faster, and with better quality. The automated block alignment makes sure precious tissue is preserved optimally. It is also a great help for making recuts, while the perfect alignment helps me produce even more slides. Trimming and sectioning are done with high precision, each section the same exact thickness. Thanks to the easy operation of the auto section, every technician in our laboratory is quickly up and running. Whatever the need, we are all confident to deliver the perfect slide each time. My name is Mark Shanti, and I'm empowered by Sakura.
Halo, kami dari PT ITS Science Indonesia. Hari ini kami akan memberikan penjelasan singkat mengenai pengujian porcine. Mengapa perlu melakukan pengujian porcine pada produk pharmaceutical? Beberapa macam bahan baku atau obat, termasuk tablet, kapsul, injeksi, dan sebagainya, dapat berasal dari produk hewani atau turunannya. Contohnya seperti gelatin, yang sering dipakai sebagai bahan utama kapsul atau stabilizer pada vaksin. Selain itu, antikoagulan seperti heparin juga dapat berasal dari porcine. Sedangkan berdasarkan Undang-Undang Nomor 33 Tahun 2014 tentang jaminan produk halal, produk yang masuk, beredar, dan diperdagangkan di wilayah Indonesia wajib bersertifikat halal. Oke, jadi bahan apa saja yang dapat mengandung porcine? Kebanyakan senyawa tersebut merupakan antikoagulan, digestive supplement seperti enzim lipase, amilase, insulin, dan sebagainya. Wah, cukup banyak ya. Tapi tenang saja, pengujian porcine ini tidak sulit kok. Ada beberapa metode yang dapat digunakan untuk mengecek kandungan porcine pada produk. Yang pertama, lateral flow device atau test pack. Metode ini cepat dan mudah, tanpa perlu banyak peralatan laboratorium yang canggih, tetapi sensitivitasnya rendah. Metode 2, ELISA. Waktu preparasinya dibutuhkan 2-3 jam. Preparasinya cukup mudah dan dapat menguji dalam jumlah banyak dalam satu kali running. Dibandingkan lateral flow device, ELISA lebih spesifik, tetapi masih memiliki batas deteksi sensitivitas yang sedang. Metode 3, real-time PCR. Metode ini adalah golden standard untuk pengujian porcine pada produk pharmaceutical dengan limit deteksi yang sangat rendah 1-5 kopi DNA. Pengujian porcine pada produk pharmaceutical sebaiknya dilakukan dengan metode PCR karena bahan baku yang diuji telah melalui proses pemurnian atau pemanasan tinggi sehingga kemungkinan yang tersisa hanya materi genetiknya saja. Selanjutnya, tahap real-time PCR ada 3, yaitu preparasi sampel, ekstraksi DNA, dan amplifikasi dengan menggunakan mesin real-time PCR. Untuk lebih jelasnya, mari melihat alat real-time PCR dari Roche, Light Cycler 96. Alat berkapasitas 96 well ini memiliki 4 channel fluorescence yang dapat digunakan sekaligus dalam satu kali pembacaan. Sebelum memulai proses PCR, profil temperatur dan protokol PCR dimasukkan ke dalam mesin. Setelah itu, tekan tombol Start. Secara perlahan akan terbentuk grafik sinyal seperti ini. Sinyal ini terbentuk dari proses denaturasi, annealing, dan elongasi yang berulang, sehingga fluorofor yang terkandung di dalam master mix dapat berpendat dan tertangkap oleh sensor mesin. Sinyal yang didapat kemudian dianalisis menggunakan software Light Cycler, sehingga terlihat level mana yang positif atau negatif. Demikian penjelasan singkat mengenai pengujian porcine dari kami. Sekian dan terima kasih.
Ladies and gentlemen, in a couple minutes, we'll start parallel session 2. And for this session, there are 5 presenters. Presenters number 17 to 21. And for this session, Dr. Ade Andriani will guide us as moderator. Dr. Ade is a senior researcher at Research Center for Biotechnology, National Research and Innovation Agency. Her research interests include microbial-based enzymes, lignin-degrading enzymes for biorefinery, and fungal bioremediation. So without further ado, to Dr. Ade, the screen is yours.
Okay, thank you for your brief introduction. Good afternoon, ladies and gentlemen. Thank you still joining with us in the parallel session 2. With the topic is food biotechnology and bioinformatics. In this session, we have 5 presenters, and they will present their topic for about 10 minutes, and then we will continue with the Q&A discussion in the final part. So please do not hesitate to ask your question in the chat box, or you can also raise your hand in the final part. Okay, to make it fast, we start with the first presenter, Bu Nuril Hidayati, with the topic promoting Setaria italica as a drought-resistant and healthy cereal for supporting food security in marginal lands. Time is yours.
Good morning, ladies and gentlemen. This presentation
is about promoting neglected and underutilized crop foxtail millet Setaria italica as a drought-resistant and healthy cereal for supporting food security in marginal lands. Neglected and underutilized species, NUS, is defined as agricultural species that are not among the major staple crops. There are some important characteristics that make NUS special and important in agricultural biodiversity that need to be conserved, developed, and promoted to poor marginal areas. One of the characteristics is adapted to local semi-arid and arid environment and tolerant to suboptimum conditions. And secondly, nutrient dense that could be useful in diversifying diets and addressing micronutrient deficiencies in poor rural communities. In Indonesia, Setaria italica can be found in several areas and is stated in Government Regulation Number 22, 2009 about food diversification. The crop can be found in some areas with different local names, such as in Sulawesi Barat is known as Tariang, and Maluku is known as Hotong, and in Java is known as Jawawut, and it also can be found in East Nusa Tenggara, Papua, and Riau. This current study is a sequel study on foxtail millet as potentially healthy food and to be developed in marginal areas. The aim of the research is to study on physiology and agronomy to improve yield and adaptability to suboptimum environment and to provide wider genetic variety by creating mutation and with gamma rays. This is the whole sequence of the research that is conducted in Research Center for Biology Brain since 2015 to 2018. Research method. Setaria seed genotype of gambir manis is radiated with different level of gamma rays, which are 0 as control, 25, 50, 75, and 100 gray. Radiation was conducted at the National Atomic Energy Agency, or BATAN. The treated seeds, which is 10 seeds of each radiation level, were planted in pots containing mixture of soil and compost, and basic fertilizer was added. Second factor was water shortage treatment. Prior to flowering stage, the plant was subjected to water shortage by reducing watering interval of every day once every 2, 4, and 6 days. The experiment was arranged in randomized block design with 4 replications. Result. Nutritional quality evaluation on 3 local Setaria italica, namely gambir manis, guru kuning, and guru merah, showed that Setaria italica can be considered as a healthy cereal. It contains about 74% carbohydrate, 10% of protein, 3% of lipid, and rich in vitamin and minerals. Agronomic properties. Plant height and leaf number can be seen in this figure that plant height and leaf number occur weekly from 2 weeks to 8 weeks after planting. We cannot see interaction between radiation and watering interval treatment. Gamma ray of 25, 50, and 75 gray show the best performance in plant height and leaf number, while 100 gray produced the most inferior performance in plant height and leaf number. There was no significant difference between watering interval of 2 days and the control. However, decreasing in plant height in plant head and leaf number was detected in 4 and 6 day watering interval. Shoot and root biomass. Gamma ray 25 gray, 50 and 75 gray show the best performance in shoot biomass. However, root biomass reduced in 50 and 75 gray, especially in 100 gray. Panicle production. There was no significant effect on gamma ray up to 75 gray on panicle numbers and panicle length and panicle weight. Significant decrease was observed in 100. Physiological properties. Physiological properties. Chlorophyll content was measured in 6 weeks after planting by using Minolta SPAD 502 leaf chlorophyll method towards fully expanded leaf, which was the third leaf from the top. The result showed that foxtail millet irradiated with 50 gray showed the highest chlorophyll content. Meanwhile, the effect of water shortage up to 6-day interval did not affect significantly on leaf chlorophyll content. Plant water potential. Measurement of leaf water potential as one of water stress tolerance indicator was carried out during vegetative growth towards the same leaf sample as chlorophyll observation by using water potential meter dew point WP4 in laboratory of Research Center for Biology. In watering interval of 2, 4, and 6 days, the control plant consistently showed the highest water potential value, followed by 25 gray and 75 gray. Meanwhile, 100 gray showed the lowest water potential in all watering treatments. Conclusion. Radiation treatment of 25 gray, 50 gray, and 75 gray perform the best plant growth. Panicle number, panicle weight, and panicle length were highest at 25 gray radiation and the lowest occurred at 100 gray. It can be concluded that optimum radiation dose for foxtail millet is 25 gray. Radiation for inducing mutation greater than 100 gray did not seem recommended for foxtail millet genotype of Cambermani. Combination of 25 gray and watering every 2 days was indicated to be the optimum for plant growth and production. It can be concluded that foxtail millet can be promoted as a healthy and tolerant crop to water shortage. Mutation by gamma ray in particular level seems possible to improve the plant towards drought resistance. Thank you.
Okay, thank you, Bu Nuril, for your interesting presentation about the Ceterica italica as alternative food, maybe for under stress conditions such as drought. Maybe to make it fast, we will continue to the second presenter, Pak Sulistyo Emantoko Dwi Putra, with the title Effect of High Carbohydrate and High MSG Intake on Body Weight and White Adipose Tissue. Time is
Hello
yours.
everybody. Today I will present my result on effect of high carbohydrate and high MSG intake on body and white adipose tissue weight. As we know that the usage of MSG in food is increased tremendously in recent years. MSG consumption in Southeast Asia is usually 2 to 3 times higher compared to the other part of the world, and ASEAN foods typically has high carbohydrate. Carbohydrate has a high glycemic index usually, and it has rapid rate of digestion and absorption, and causing hyperglycemia and make rapid hungry feeling, whereas MSG intake can lead to overeating effect. Combination between hungry feeling and overeating effect in the long term will lead to obesity. In this study, we perform 4 groups of animal experiment called as control, high carbohydrate diet, high MSG diet, and combination between high carbohydrate and high MSG diet. For the control, the feed of the animal experiment contain 57% of carbohydrates. For high MSG, we give 10% of MSG in their diet. For high carbohydrate diet, we give 71% of carbohydrate in their diet. And for the combination, we give 71% of carbohydrate and 10% of MSG. During the experiment, animal will get feed and drink at libitum. They also placed in a solitary in one cage, and they maintain at room temperature. And they got the same duration of light and dark. The experiment was performed in 14 weeks, and afterward, we sacrificed the animal. This is the result of the experiment. As you see, in the beginning of the experiment, there are no difference of body weight between control, high carbohydrate, high MSG, and combination diet. After week 2 of the experiment, we also did not show any difference of body weight among the group, the experiment. After week 4, we got that the body weight of high carbohydrate diet is higher than control. Also, we saw the same phenomena for the combination of high carbohydrate and high MSG diet. Also in week 6 of the experiment, where the body weight between group is different. High carbohydrate, high MSG, and the combination have heavier body weight compared to the control. We continue the experiment for week 8, 10, 12, and 14. At the end, we show that the high carbohydrate diet has heavier body weight compared to the control, but not with high MSG and the combination of high MSG and high carbohydrate diet. How about the weight of white adipose tissue? As you can see in this picture, that the adipose tissue of high carbohydrate diet is heavier than control, and also the white adipose tissue of the combination diet is heavier compared to the control, but not weight of white adipose tissue from the high MSG diet. For future direction, we plan to do a research on epigenetic regulation. Why the white adipose tissue of high carbohydrate and the combination between high carbohydrate and high MSG is heavier compared to the control. Why the weight of white adipose tissue is almost the same between control and high MSG diet. So as conclusion of for high carbohydrate diet, we can say that this diet is a potent obesogene due to increased white adipose tissue mass and lead to the most significant weight gain. But for high MSG intake, we can say also high intake of MSG as an obesogene agent. You can see for the combination high carbohydrate intake and high MSG intake, combined intake of high carbohydrate and high intake of MSG lead to obesity. Underlying mechanism needs to be investigated further. Thank you for your attention.
Okay, thank you, Bapak Sulistyo, for your interesting presentation. And if you have questions to all participants, please write in the chat box. Okay, we will continue to the third presenter, Budiah Ayu Widyastuti, with the title Ethanolic Extract of Parkia speciosa peel, its effect to alkaline phosphatase and catalase level of Wistar white red with jelantah exposed. Tan is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Thank you for the opportunity for me to present our study. My partner and I come from Universitas PGRI Semarang. Our study is about ethanolic extract of Parkia speciosa peel, its effect to alkaline phosphatase and catalase level of Wistar white red with jelantah exposed. Sorry. Okay, the Indonesian habit to consume fried food with repeated use of palm oil, as we know, we call it jelantah, will decrease nutritional quality, then causes oxidation, and oxidation will generate free radical compounds, then trigger dyslipidemia. The dyslipidemia in our body will lead to organ damage, include liver, kidney, heart, blood vessels, and others. The free radicals from jelantah leads to liver damage, which marked by the increase of alkaline phosphatase level. The ALP level increased gradually due to the severity of liver damage. Free radicals from jelantah also induce oxidative stress, which involve preventive mechanism, repair mechanism, physically defense, and antioxidant defense. For antioxidant defense, it involves several enzyme activity, including CAT or catalase. Based on that fact, development of natural product as please reducer of jelantah trait is necessary. One of the candidates to be developed as reducer of jelantah trait is the type or Parkia speciosa, especially from the peel. The objective of this study is to find out the alteration of alkaline phosphatase or ALP and catalase or CAT level of Wistar white red, which induced by jelantah and treated by ethanolic extract of Parkia speciosa peel. For the methods, the sample preparation, the Parkia speciosa peel was sliced, then dried, and then mashed and shipped with 70 ml. For the extraction, the powdered peel was macerated with 500 milliliters of ethanol for 24 hours. The filtrate was collected and concentrated with rotary evaporator at 45 degrees Celsius. Then, the in vivo test, we used 4 groups of Wistar white reds. With group 1 is group with no treatment. For group 2 is the group with 1 milliliter jelantah treatment plus 100 milligram per liter extract of Parkia speciosa peel. Then for group 3 is group with treatment 1 milliliter jelantah with 200 milligram per liter extract of Parkia speciosa peel. Then for the group 4 is the positive control with only treatment of 1 milliliter jelantah. Okay, after 28 days of the treatment, the serum of the Wistar white red is collected and then analyzed with ALP and CAT analysis with dialysis kit. The last phase of the method is statistical analysis. We use SPSS 17 version. For the result, Table 1 showed the group 4 as positive control has highest alkaline phosphatase level because the Wistar white reds in group 4 were exposed by jelantah 1 milliliter jelantah for 28 days without treatment of Parkia speciosa peel extract. Those jelantah exposure leads to liver abnormality marked with enhancement of alkaline phosphatase level. The lowest alkaline phosphatase level was shown by group 1 as negative control with no treatment. In group 1, Wistar white reds, did not treated by either jelantah or ethanolic extract of Parkia speciosapia. For group 2, showed the higher alkaline phosphatase level than group 3. The lower alkaline phosphatase level in group 3 indicate the potential of ethanolic extract of Parkia speciosapia to reduce negative impact of jelantah exposure. Then for the catalase level of visceral white rates with jelantah and ethanolic extract of Parkia speciosapia treatment after 28 days showed that among 4 groups, group 1 as negative control has the highest catalase level because visceral white rates in group 1 were not exposed by jelantah, and visceral white rates which exposed by jelantah then treated with ethanolic extract of Parkia speciosapia has lower CAT level than which not treated with ethanolic extract of Parkia speciosapia. Catalase level of group 2 and group 3 are 3.81 and 5.17 respectively. Group with lowest CAT level is group 4 as positive control. Treatment of ethanolic extract of Parkia speciosapia show a significant difference of CAT level at group 2 and group 3. The higher dose of ethanolic extract of Parkia speciosapia treated with steroid rates enhance CAT level produced by the cell, so it prevents cell damage. The lowest CAT level at group 4 as positive control show that jelantah has negative impact for the cells due to its ability to enhance free radicals activity at the body. On the other hand, the highest CAT level at group 1 as negative control show that although CAT level at group 2 and group 3 are higher than group 4, but it cannot be higher than group 1 because visceral white rates at group 2 and group 3 were exposed by jelantah before treated by ethanolic extract of Parkia speciosapia, while group 1 were not. Treatment of ethanolic extract of Parkia speciosapia enhanced CAT level after visceral white rates were exposed by jelantah, but never been as high as which not exposed. The conclusion of this study is jelantah exposure enhanced ALT level, which can be reduced by ethanolic extract of Parkia speciosapia, then visceral white rates which exposed by jelantah then treated with ethanolic extract of Parkia speciosapia have lower ALT level than which were not. The higher dose of AFPP, the lower ALT level. Jelantah exposure reduced catalase level, which can be enhanced by ethanolic extract of Parkia speciosapia. Visceral white rates with jelantah exposure then treated with ethanolic extract of Parkia speciosapia have higher CAT level than which were not, and the higher dose of ethanolic extract of Parkia speciosapia, the higher CAT level. This is some of our references. Thank you for your attention, and wassalamualaikum warahmatullahi wabarakatuh.
Thank you for your interesting presentation about the ethanol extract of potash against the free radical of jelantah. So please do not hesitate to ask the question related to this interesting topic in the chat box. Okay, now we continue to the fourth presenter, Bapak Sugiono Saputra, with the title Exopolysaccharide Production and Amylase Activity of Lactic Acid Bacteria Strains. Time is yours.
Hello everyone, my name is Sugiono Saputra from Research Center for Biology, BRIN. Today, I'd like to present my research with the title Exopolysaccharides Production and Amylase Activity of Lactic Acid Bacteria Strains. So, bacterial exopolysaccharides, or simply just EPS, have been widely described since many years for their beneficial applications, as well as their uniqueness in structural diversity. They're actually sugar-based parts of natural polymers secreted by microorganisms into the environment. They have various roles, including enveloping the bacterial surface, protecting bacteria against environment, signaling molecules, as well as additive agent to form biofilm. And then because of their roles, EPS have potential for application in several fields, such as textiles, cosmetics, bioremediation, food, and also therapeutics. EPS might exist as homo and heteropolysaccharides and can be produced from lactic acid bacteria strains, or are widely known as probiotic bacteria. And EPS produced by lactic acid bacteria are known for multiple bioactivities and health promoting effects, such as immune modulatory, antioxidant, anti-allergenic, and also protecting against infectious agents. And there are common requirements to screen the probiotic bacteria from several habitats and sources. The common include low pH, bile salt, antibiotic sensitivity, addition, and also bacterial production and aggregations. And in this study, we're focusing on two aspects, including the EPS production, and also the second one is amylolytic activity. Why? It's because the first EPS, they have potential for therapeutic applications, and by adding this character to probiotic screening will be very useful for giving plenty of beneficial properties to the microbial communities as well as the gut health. And then for additionally, for the enzymatic activities of probiotic bacteria, it's also crucial to assist food digestion by catalyzing the hydrolysis of starch into oligosaccharides, which is known as prebiotics. And then when adequate amount of prebiotics produced during the starch digestion, it will also provide food for the good bacteria, and it will suppress the colonization of bad bacteria. And then because of the two aspects of those probiotic properties, in this study we aim to examine a collection of LAB for their potential EPS producers as well as the amylase activity. In this study, a collection of unidentified human lactic acid bacteria from Microbial Cell Biology Research Laboratory at Research Center for Biology is used. And then for the EPS production and amylase production, LAB bacterial strains will grow in 5 milliliters lactobacillus amaris broth, which with two different carbon sources, including sagu starch and glucose. The modified amaris broth were inoculated with 5% of pre-grown amaris medium culture, and then the cell was harvested after 72 hours for EPS production, and then 24 hours for amylase productions. And then for the EPS, the supernatant obtained from centrifugation was dialyzed by dialysis tubing from cellulose membrane with molecular weight cutoff greater than 12,000. So as you can see here, this picture, so basically the dialysis is a biochemical process that allows preparation of small molecules, which is possibly small saccharides from mixed different sizes of molecules. So after the process, the dialysis, the small molecules diffuse through the pores into the solvent, and the bigger molecules or the polysaccharides remain in the membrane. Then the EPS yield measured using the phenolsulfuric acid or amylase activity was measured using starch iodine assay. So a total 7 out of 18 LAB strains were successfully revived and well grown in sago starch. And then all selected 7 isolates were gram-positive and morphologically similar with the coccus and moist and mucoid character. And as you can see here, for the APS yield, glucose was an excellent substrate for abundant APS synthesis, produced more than like all the isolates, more than 200 milligram per liters with the 2 isolates, MEE 02 and MK03 gave the high result. And then all of the isolates were identified using 6-kinase RNA analysis. And then, as you can see here, all of them belongs to 3 genus, namely Enterococcus, Leconostoc, and also Streptococcus. And comparing to the other study, there are numerous bacteria that claim as probiotic bacteria, which is including Enterococcus faecium, which is also isolated from the feces with EPS yield reaching up to 23 gram per liter of exopolysaccharides. And then there is evidence as well that Enterococcus faecium and also Enterococcus faecalis, they also produce amylase enzyme, and then they actually were isolated from fermented food. And the current study here also shows that Leconostoc species, they are able to produce amylase, and then with comparison with the Leconostoc mesenterides, they specify its alpha amylase activity. So in this study, we have at least 2 findings. First, the highest EPS yield in glucose and sago starch medium was obtained by 2 isolates, Enterococcus faecium, ME02, and Enterococcus faecalis, ME03. However, for probiotic bacteria screening, these 2 isolates possibly need further evaluation as probiotic bacteria due to they may contain virulence factor. And then the second finding is that Leconostoc lactis, MC021, they regarded as generally recognized as safe or GRAS microorganism, which is maybe can be further developed and potential as probiotic bacteria. That's all from me. Thank you.
Okay, thank you, Dr. Subiana Saputra, for your interesting presentation regarding to the lactic acid bacteria and their potency to produce amylase activity. Okay, let's continue to the last presenter, Bu Tati Kusniati, with the title Reducing Sugar and Total Acid Concentration of Vegetable Yogurt with Addition of Amyloproteolytic Enzyme Lactobacillus fermentum EN 17-2. Time is yours.
Ladies and gentlemen, I would like to present our research result, reducing sugar and total acid concentration of vegetable yogurt with addition of amyloproteolytic enzyme Lactobacillus fermentum EN 17-2. Vegetable yogurt can be consumed by all humans, and it can be used as alternative drink, especially for people with lactose intolerance. In addition, yogurt vegetables have high carbohydrate and protein content. Vegetable ingredients that can be used as alternative can come from the nut and mushroom. Therefore, there is a need for modification to hydrolyze carbohydrate and protein in vegetable material into simple form. One way of modifying carbohydrate and protein by adding amyloproteolytic enzyme to phosphor from bean and mushroom. Several lactobacillus bacteria known to be able to produce amyloproteolytic enzyme, including Lactobacillus lactis, Lactobacillus plantarum, Lactobacillus rhamnosus. Previous study report their juice from extract of phosphor from bean and mushroom with addition of amyloproteolytic enzyme Lactobacillus subsunensis AM3832 was processed through a fermentation process produce a product in the form of vegetable yogurt. The study focused on reducing sugar content and total acid of vegetable yogurt with the addition of amyloproteolytic enzyme Lactobacillus fermenting AM172. The method used fruit media and production of amyloproteolytic enzyme by using an NS fruit and NS starch and nutrient fruit material. Subculture of Lactobacillus fermentum by using NS fruit media incubated at 37 for 24 hours. Protease production by using centrifugation and then supernatant of time. Protease activity by using Horikoshi method. One protease unit is defined as the number of milliliters of protease required to produce 1 tyrosine per minute using casein as substrate. For vegetable paste flour, peanut or mushroom flour with distilled water, heat at 70 degrees Celsius. Add 1 unit protease incubated at 37 24 hours. Determination of degraded protein by using Nielsen method. Cross-banded protein degrade for semi-protein cross-conversion factor. Amylase protection by using centrifugation and amylase activity. The formula is glucose concentration plus 1 percent plus diffusion factor divided molecular weight glucose plus incubation time. For vegetable yogurt, juice extract from vegetable phosphor with enzyme extractor for fat incubated 34 degrees Celsius for 5 hours or for 18 hours at room temperature. Reducing sugar by using Benza method with formula sample concentration minus control concentration divided sample concentration of 100% as reducing sugar. Total acid analysis by using AUAC method and statistical analysis by using analysis of variant with true repetition and further analysis by Duncan analysis. The result and discussion show that table 1 alpha amylase and protease activity of L-fermentin, the enzyme activity of alpha amylase was significantly different with protease. The difference in the activity of alpha amylase and protease due to the different character of each enzyme. Amylase hydrolase amylase, while protease hydrolase protein. Compared to degraded protein content of bean and mushroom base flour with protease. So that degraded protein concentration from soybean base flour and mung-bean base flour is not significantly different. Also, in oyster base flour and button base flour is not significantly different. but two of bean phosphor and mushroom phosphor was significantly different than homemade water. The higher concentration of protein in fish pasta flour with protease than in water, and the lower concentration in mushroom phosphor was thought due to nutrient substrate required for protease activity, and nut phosphor was better than in milk water, and in mushroom phosphor was smaller. Factor 3, reducing sugar content of bean and mushroom phosphor with alpha amylase. The concentration of soybean phosphor was significantly different than pumpkin phosphor, but for oyster phosphor was not significantly different than cotton phosphor. Both bean phosphor and mushroom phosphor was significantly different than homemade water. The low concentration of reducing sugar in phosphor from nut and mushroom with alpha amylase compared to water made, probably due to the presence of inhibitor and cofactor in pasta flour and milk water. Therefore, reducing sugar content of yogurt based on fish extract from bean and mushroom pasta flour with addition of amyloproteolytic enzyme. The concentration of reducing sugar of soybean yogurt was significantly different than pumpkin yogurt, but oyster yogurt was not significantly different than cotton yogurt. But both bean yogurt and mushroom yogurt was significantly different than milk yogurt. The reducing sugar concentration and amyloproteolytic bean yogurt and mushroom yogurt lower than whole-milk yogurt because the reducing sugar content in milk water was higher than in nut phosphor and mushroom phosphor as shown in table 3. Table 5, total acid content of yogurt with juice, flour, bean, and mushroom paste with addition of amyloproteolytic enzyme. The percentage of total acid concentration soybean yogurt is not significantly different than pumpkin yogurt, and also oyster yogurt was not significantly different than cotton yogurt. But both was bean yogurt and mushroom yogurt were significantly different than milk yogurt. Total acid concentration in amyloproteolytic nut yogurt and mushroom yogurt lower than milk yogurt. This was due to the reducing sugar content in amyloproteolytic nut yogurt and mushroom yogurt lower than in amyloproteolytic milk yogurt as shown in table 4 before. So the conclusion is research and reducing sugar content and total acid of vegetable yogurt with amyloproteolytic enzyme L-fermented AM17-2 has shown that the reducing sugar and protein degradation of enzymatic vegetable phosphor has been detected. The highest reducing sugar concentration of enzymatic vegetable yogurt is found in soybean yogurt. Total acid of enzymatic soybean and enzymatic bean yogurt higher than oyster and cotton yogurt. For reducing sugar and total acid of pomade higher than vegetable milk. The total acid content of amyloproteolytic vegetable yogurt was included in the range of total acid content determined the Indonesia national standard. Based on reducing sugar and total acid content, soybean yogurt was close to milk yogurt than the other vegetable yogurt. So, soybean yogurt was better than the other vegetable yogurt. Thank you.
Okay,
Thank you, Professor Tatik, for your interesting presentation. Okay, ladies and gentlemen, now we continue to the Q&A session. We have already 2 questions in the chat box. For the first is from Pak Elof Rifki from BRIN. He asked to Mrs. Dia Ayuwidya Susti, is there any specific reason for using the peel of Parkia speciosa instead of the fruit? And what about the other related plants with Parkia speciosa in Leguminosae, such as Leucaena, Leucopeta? Do they possess the same potential in ALP and cut activity? Thank you. Please, Mrs. Dia Ayuwidya Susti.
Thank you for the question. Let me introduce myself. My name is Papa Nurdiansyah. I'm as the second author. And here I represent Mrs. Dia Ayu to will answer the question from Pak Elo. I think we use the peel Parkiosa peel because the peel of Parkiosa speciosa is only remain as trash with no function, even though it has some potential with its biochemical activities, such as flavonoid, phenolic content, and others. And how about the other related plant like Leucaena? I think other plant or Leucaena may has some potential, but it may differ in the level of or antioxidant activity due to its different bioactive content or compound in the Leucaena. I think it's.
Thank you. Which is, can I ask also, which is higher, the fruit and the peel in the active metabolite?
Oh, the higher content based on the study is higher in peel than in a fruit of Parkiosa species.
Okay.
Okay.
Thank you.
Thank you.
Okay, next we continue to the other question for Dr. Sugiono from Bu Ai Hertati from BRIN. She asked, why did you isolate LAB from human feces? Is there any specific reason? In the IBD research, there is a developed research about using normal human feces to IBD patients in order to make some gut microbiotas in normal pattern. Thank you very much. Please, Dr. Sugiono. Is Dr. Sugiono attend?
Oh yeah, sorry.
Okay.
Thank you.
Please, Dr. Sugiono.
Okay. That's a good question, Ibu Ai. So basically, this is a part of a bigger study, which is we aim to obtain exopolysaccharide producers from various sources, and we want to use sago starch as a local source material to produce bacterial exopolysaccharides. And because it's for human, we tend to obtain the bacterial species from human too. And we thought that it's highly likely if there will be various species of bacterial exopolysaccharide producer from an environment which is adapted for this diet. So we isolated the bacteria from human who consume a high starch diet, which is we conducted this isolation in Maluku. It's well known for sagu starch is the like a staple food in that area. So they consume sagu starch diet every day, and then we know that they also have a good benefit to the gastrointestinal tract for them. And yes, that's the reason why we conducted bacterial isolation from human feces.
Thank you, Dr. Sagyana Saputra, for your clear answer. Okay, we have 9 minutes left for Q&A session. If you have a question, yes, Dr. Fahru Roji, please.
Okay, thank you, Dr. Ade, for the opportunity. So I will ask my question directly to, I will address to Prof. Kusmiati, to Prof. Kusmiati. Okay, so hello, Bade.
Okay.
Okay. I will directly deliver my question to Prof. Hermiati. So my question is, I don't follow your presentation fully, but if I'm not mistaken, you mentioned that you add the amylase enzyme and also protease enzyme in the yogurt. So what do you mean by amyloproteolytic enzyme? So mean you put all together, and how much the percentage of the amylase and also proteolytics. And then the second question is, what the advantage of the adding amyloproteolytic enzyme into the yogurt products? Thank you, Dr. Ade.
Okay, thank you, Dr. Fahru Roji. Maybe Professor Tatik, you could directly answer the question from Dr. Fahru Roji, please.
Okay, thank you very much for your question, Dr. Fahru Roji. Actually, I put amyloproteolytic enzyme, amylase and protease separate, not mix. For the paste mushroom and bean paste flour. The end of why we put amyloproteolytic enzyme, actually paste flour from bean and mushroom as compared with wheat flour is not so homogeneous. So to be better for degradation of protein and amylase. So we put amyloproteolytic enzyme to make more homogeneous. So to make easier to digest as yogurt, put amyloproteolytic enzyme. Is it okay or not
Okay, okay, yeah, okay.
Is it okay?
Yeah, it's clear, Prof. Tatik. But one more question: these two enzyme was produced by Lactobacillus fermentum, Prof. Tatik?
Oh yeah. And this amylase and protease isolated from Lactobacillus fermentum from Enggano Island.
Enggano Island, but
From Mira from Enggano Island.
But they can produce two enzyme directly, amylase and protease.
Yeah.
Okay, thank you.
Oh, okay, thank you very much.
Okay, thank you, Professor Tatik and Dr. Fahru Roji for nice discussion. Okay, we have 5 minutes left for Q&A. Please raise your hand if you have any question. Maybe I have, if there's no question. I have question for Pak Sulistyo M. Mantoko Dwi Putra. Pak Sulistyo, is there any scientific suggestion to our daily diet in consumption carbohydrate and MSG to our healthy daily diet regarding to your research result? Please, Pak Sulistyo.
Thank you for the question. Let me introduce myself. My name is Fariski. I represent Mr. Mantoko because he has another business. Please repeat the question, Dr. Ade.
Yes, regarding to your result, is there any suggestion how we could consume carbohydrate and monosodium glutamate in our daily diet?
Yes, absolutely. In MSG, 1 gram per day 1 gram per day per kilogram berat badan per body weight. And for carbohydrate, it's like 50% until 60% carbohydrate in daily intake.
So regarding to your result, monosodium glutamate could help our diet program.
Yes, but MSG have effect in gastrointestinal like ulcus like erosion in the epithelial gaster and small intestine, and it's make absorption disturbance like this.
Okay, thank you for your answer. Okay, now we have, if there's no any question, I have question, the last question for Bu Nuril Hidayati about the Setaria italica. Is there any drought condition affected the nutrient content in the Setaria italica? Please, Bu Nuril Hidayati. Bu Nuril Hidayati still attend the seminar? Okay.
All right. I'm sorry.
Okay. Okay.
Yes. Actually, Setaria italica has been already known as a drought-resistant plant. And in the environment, natural environment, it can be grown in the marginal and rain-fed condition. But in here, we need to be optimum condition for high yield, because in the environment, natural environment, it still be grown as underutilized, and the yield is still very low, about 3.5 to 4 ton per hectare. But in here, we need to obtain optimum condition for water condition and to increase yield to get the optimum yield and optimum water condition.
How about the nutrient content is affected by the mutation, gamma mutation, or?
The nutrient content, the effect of gamma radiation to nutrient content, unfortunately, we did not analyze for each treatment of gamma radiation, but maybe next we will try to evaluate the nutrient contents in the gamma treatments.
Okay, thank you, Bu Nuril, for your answer.
You're welcome. Thank you.
Okay, ladies and gentlemen, we have been finished all the presentation for all presenters. I think we finished the session. Thank you for joining us. Don't forget to join in the other session. Thank you.
Ladies and gentlemen, thank you very much for your participation. Thank you to Dr. Ade and all presenters for sharing the knowledge. We'll have 10-minute breaks and we'll start again at 4 p.m. Thank you. Ladies and gentlemen, before entering session 3, we'll enjoy video from our partners. Before start. Okay, ladies and gentlemen, we'll start parallel session 3 with moderator Dr. Renat Fahrurozi. Good afternoon, Dr. Fahrurozi. Thank you very much for your time. And Dr. Fahrurozi is a researcher at a Research Center for Biotechnology, National Research and Innovation Agency. His research interests include the development of starter culture, as well as enzymes and microbial cell factories for food applications. Okay, without further ado, to Dr. Fahrurozi, the time is yours.
Okay, thank you very much, Ms. Eva, for the introduction. So this is the last session of this today symposium, and it late of afternoon. Perhaps that you are not feeling sleepy, because maybe you can take first a coffee break with you, and then because we still have 4 interesting presenters for today. The first one was from the Miss Marta Sari from Research Center for Biotechnology, and the second from Miss Uri Perwitasari from Research Center for Biotechnology, and the third from Miss or Mr. Rizamel from the University of Tanjung Pura of Pontianak, and the last one will be delivered by Elo Rifki Firdiani from Research Center Biology, maybe from BRIN. So I think the rule of this symposium, so all of the presenter will have about 10 minutes of video presentation, and then we will have Q&A by in the end of this all presenter about 10 minutes, and then so I will make sure all of the presenter is already in room. So Miss Martasari is already here, Miss Puri Perwitasari, but number 24, I think it's not yet in the rooms. Could you, the organizing committee, could you please invite into the rooms?
Yes, we'll try to track
Number
presenter
24,
number 24.
Mrs. Elo Rifki is already also in the room. Or there is some replacement for Mrs. Rizamel, maybe the not replacement. I mean the others presenter in the same topic.
No, she was present in session 2. Maybe
Okay.
she hasn't come back from break.
Okay, so meaning we have only 3 presenter in this session. So to save the time, I think we will start with the first presenter from Miss Martasari, and please the organizing committee to apply the presenter number 22.
Welcome everyone. Assalamualaikum warahmatullahi wabarakatuh. Thank you for your time and chance to me to present on the International ISIBIO Symposium 2021. Previously, I would like to introduce myself. My name is Martasari from Research Center for Biotechnology, BRIN. And this time, I would like to present our study, and the title is The Comparison of Meningogenetum Genemon Linseed Flora and Extract from Industrial and Native and their Activity Test as an Antioxidant. And the following names are members that support our study. Introduction. As we know that Meningogenetum genemon found many tropical area including Indonesia, member Genetum family and Genetaceae species as food traditional as well as folk medicine, with high source of nutrition and wide range of health benefits. The genetum seed material possesses physiological properties like antioxidant, antimicrobial, enzyme stimulant, antihypertense, anti-cancer, and hormone analog. According from other studies state that the seed extract of Genetum genemonus contains stilbenate compound consisting of Genemonocida A, C, D, Genetin C, and resveratrol can act as various radical scavenging activity. And our focus research to investigate the antioxidant from the part of seed Genetum genemon. And the aim of the study to compare the antioxidant activity, including total phenol, total flavonoid, and to identify compound profile from flora and extract genemon seed from industrial and native. And our methods as follow. This research was conducted in January until June 2021 at Laboratory CD3 Research Center for Biotechnology, BRIN. Starting from pre-processing sample of seed genemon, we by using maceration with 70% ethanol. We use 3 samples, namely sample A is called seed extract from Industria, sample extract. And Furthermore, we check the qualitative phytochemical tests and the quantitative antioxidant ABTS activity, including total flavonoid and total phenol. And the analysis of profile of major compound with HPLC and interpretation of chemical compound by GCMS. Result and discussion. The result of phytochemical assay of seed gnemon qualitatively are presented at table 1. And based on table 1, that sample A contains flavonoid, saponin, steroid, triterpenoid compound, while sample B and C contain flavonoid, saponin, tannin, steroid compound with the reasonable color strong intensity. The plain phytochemical natural response has guided particular attention because it provides great benefits human health. It's study from Mehta et al. 2010. Next, the determination of total phenol, total flavonoid, and ABT's antioxidant activity level from gnemon seed were also seen on graphic 1. And based on the graphic 1E, show that the higher of total phenol was found on sample E, which is 309.36 milligram gallic acid per gram sample, higher than sample B and sample C. And the result of the total flavonoid content can be seen on graph 1B. Its sample E as big as 22.55 milligram quercetin per gram sample, higher than sample B and sample C. And the strength of antioxidant activity on graphic 1C show that sample A has has strong antioxidant activity as big as 89.67 ppm, higher than sample B and sample C. Because sample E obtained on IC50 value less 100 ppm, but the standard more higher than others. The IC50 value are used for antioxidant classification, and the lower of IC50 value indicates the higher antioxidant activity. A study from Rosa et al. 2020. Next, the analysis of profile compound of seed gnemon by HPLC. And the characterization of the industrial and native oat extract and flour from seed gnemon has been done in figure 1. And based on figure 1, the sample absorbance patterns and retention time is reviewing seed gnemon generally have the slightly some configuration at least 3 patterns. And dominant peak that are detected in retention time of 4.05 minutes and 26.13 minutes and 29.32 minutes. These findings are consistent with the literature that melinjo seed extract successful was identified some major stilbenate compound, a study from Kato et al. 2009. Next, the sample identification of chemical compound from seed gnemon search results are represented on table 1. And based on table 2, started from the dominant spectrum based on at least similarity index of 90 until 95% from NIS1 database. And content from sample E is composed 4 main component, sample B for 1 main component, and sample C composed 2 main component. The constituent chemical component of the extract, both native and industrial, and melinjo seed powder contain hexadeconic acid with varying similarity value. And the antioxidant effect of hexadeconic acid from brown alga from Turbinaria ornata was evaluated by the PPA test with IC50 70.62 microgram per milliliter and superoxidized radical scavenging with IC50 71.4 1 microgram per microliter respectively, a study from Barats et al. 2021. And our conclusion is follow. The sample A from industrial seed melinjo consists of phytochemical compound of flavonoid, saponin, steroid, and triterpenoid, while sample B and C contain flavonoid, saponin, tannin, and steroid with the reasonable color, strong intensity. And the antioxidant activity of melinjo seed extract from industrial sample A have the potential antioxidant with ABT score with IC 50 value 97.67 ppm, total phenol 309.36 mg per gallic acid per gram sample, and total flavonoid 22.55 mg per cent per gram sample is compared with the sample B, sample C. All samples have similar Mayer compound profile at least 3% by HPLC and predominantly detected in retention time of 4.05 minutes, 26.13 minutes, and 29.32 minutes. Example E, B, and C have the same spectrum composition by GCMS spectrum that contain and hexadeconic acid with different similarity index value based on NIS1 library database. And this occasion, the author expressed thanks to the Deputy of IPH BRIN for funding support from the DIPA PM 2021 project. And thank you for all.
Okay, thank you very much, Ms. Martasari, for your nice presentation. And then for answer and question, we will do in the end of this session, and then we continue with the second presenter, Ms. Urip Perwitasari, entitled of submergent fermentation of cacao pod hulls for citric acid production by Aspergillus niger. So you have 10 minutes for presentation, and then time is yours, please.
Thank you for the opportunity. Let me introduce myself. My name is Urip Perwitasari, and I will present with the entitled Submerged Fermentation of Cacao Pod Husk for Citric Acid Production by Aspergillus niger. Citric acid production has many functions in food industry. Citric acid as antioxidant and pH stabilizer. In chemical industry, citric acid as a cleaning agent in metal removal. In addition, citric acid is a macronutrient for a plant, so agricultural industry add this chemical into fertilizer. Pharmaceutical industry also needs citric acid as anticoagulant in drugs. In production citric acid, there are 4 ways to produce citric acid. First, by fat extraction, and the second by chemical production, third by fermentation with a glucose as a substrate. But nowadays, the fermentation utilize a biomass as a substrate. Therefore, in this research, we utilize cacao pod husk from cacao agro industry. Carbohydrate in cacao pod extract can be alternative carbon source for microbe to produce citric acid. Therefore, citric acid produced can be sustainable. And also, citric acid by submerged fermentation with Aspergillus niger can be produced, so industrial and environmentally friendly can be created. In this research, we using response surface methodology with box Banken design for the analysis method, and we use design expert 10 edition for the software. This research using 3 factors: concentration of cacao pod husk, nitrogen concentration, and incubation period. We also adding ethanol and molasses waste as inducer in methanol in medium. And then we use Aspergillus niger from Indonesian culture collection in ACC. And the results show, this is the graph relationship between concentration of cacao potash and nitrogen. It is show that cacao potash has upward trends, which indicate the optimum point to produce is the highest concentration of cacao potash for citric acid production. And the y-axis, it is have the curve show that the surface to be convex, but the decreases at the top and the bottom, which means that the optimum point to produce citric acid is in the middle of concentration nitrogen. Okay, and the second one is relationship between concentration of cacao potash with incubation day. This is has same pattern as figure 1. The long fermentation allow to conversion of biomass to citric acid to be more significant because the number of microbial cells increases with increasing fermentation time. However, if the substance is diluted, the concentration of citric acid will be decreased because citric acid will used for metabolism of microbe if the sugar concentration in medium is diluted. And figure 3 show relationship between nitrogen concentration and incubation time. The result indicate there is no interaction between nitrogen concentration and incubation time. This can be proven by the p-value in the interaction in next slide. Okay, this is the ANOVA. The ANOVA of the response to the citric acid concentration. Based on p-value, the model has p-value not more than 5%. It is indicate that this model is significant for citric acid production. And the lack of it of this model is higher than 5%. It is indicate the lack of it not significant in citric acid production. And the factor cacao potash and nitrogen concentration has a significant factor in production of citric acid. On the other hand, the incubation time has insignificant factor for citric acid production. In addition, the relationship between nitrogen and incubation time also has insignificant factor for citric acid production. And the verification model, the software recommended that concentration of cacao potash is around 10%, and for nitrogen concentration 0.3 time incubation will predict about 1 1,900 ppm of citric acid with the desirability value around 0.8%. It is show that there is a possibility of error in citric acid production about 14%. And the verification result or the actual value for the citric acid production in this research, the concentration of citric acid is around 1,900 ppm of citric acid. And the difference between the predict and the actual value is about 95 ppm with the p-value a great bigger than 5%. It is indicate that predicted and actual value of citric acid production are not significantly different. We also compare citric acid production from the other biomass. It can be seen that the pretreatment method affect the production of citric acid. Biomass with chemical, also biological pretreatment can produce higher citric acid than only physical pretreatment. However, cacao potash can produce citric acid higher when we compare with rice straw and fruit pulp. This biomass also have some pretreatment. They are only drying and grinding the biomass. And for the conclusion, the optimum condition for producing citric acid with submerged fermentation suggests by design expert is for the substrate concentration around 10% and nitrogen concentration around 0.3%, incubation days for 5 days and 10 hours, and it predict the citric acid production around 1,900 ppm. It has same result with the verification model. With the verification, it's around 1,900 ppm of citric acid. However, the additional pretreatment method is needed for cacao potash waste to remove the lignin from cacao pod. I think that's all, and thank you for the time. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Mrs. Uri Perwitasari, for your nice presentation and good idea. I think please keep your question for the participant, and you can ask by the end of all of this session. And for the next presenter, should be from number 24, but since they are not in this room, so we continue with the presenter number 25, Mrs. Elo Ripki Firdiani, entitled of Anti-Coronavirus Potential Activity of Phytochemical Contained in ISIBIO Kunz in Silico Molecular Docking Study. I think it's very interesting also title. So please, Mrs. Elo, for your time, the 10 minutes.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. In this moment, I would like to present our work entitled Anti-Coronavirus Potential Activity of Phytochemicals Contained in ISIBIO Kunz through in silico molecular docking studies. We are a team of 3 consisting of Lois Nawati Anu, Aga Ranjana, and myself, Ella Kiperdiana. We are from National Agency of Research and Innovation. In specific, Biodiversity Garden is a center for plant conservation and botanical. As we all know that COVID-19 has become a pandemic, including in Indonesia. It had already infected more than 4 million people as of 13 October since its first case reported. Surely, there are a lot of efforts to overcome this, but exception to find an alternative treatment. On the other hand, plants have shown to possess phytochemicals with antiviral activity, one of which is ISIBIO. Unfortunately, this potential theory has not been widely studied. For this purpose, we use in silico molecular dating, in which it is an initial method used for screening drug activity. A particular compound is interacted towards specific protein target responsible for the occurrence of disease. ISIBIO is one living collection of probability garden to be reviewed for its benefits, especially as medicines against coronavirus. Therefore, the study was conducted to determine the anti-coronavirus potential of phytochemicals of ISIBIO by in silico molecular dating. We conducted this work through 3 steps. First of all, we determined the phytochemicals of ISIBIO, where we obtained this information in knapsack website. Then, we traced their 3D structures in FATCHAIN. Then, we determined the target proteins responsible for coronavirus infection through literature studies, and we ascertained their structures according to NCBI and found their 3D model via Swiss model. And finally, we screened this anti-coronavirus potential activity using protein molecular dating utilizing PR expression 0.8. The docking data were then analyzed using PyMod 32-bit version 1.3.0.0 and visualized via protein plus. Here are the results and discussion. We found out there are 6 compounds in ISIBIO having the potential as anti-coronavirus. There are ampreloxetine, naringenin, miricetin, rubinetin, pisiatenol, and resveratrol with their virus activity properties. Then we also found that there are 3 main proteins involved in coronavirus infection. There are 3-CL-PRO, PL-PRO, and ACE2. 3-CL-PRO has a role in coronavirus replication, while PL-PRO has a role in coronavirus replication and host immune response appointing, while ACE2 has a role to be a receptor for spike protein of coronavirus. After interacting the compounds in the proteins, we found out that all the compounds tested have interaction with all the proteins tested. But the strongest activity shown by miricetin with 3-CL-PRO with binding affinity of 8.1, and rubinetin had the strongest activity with PL-PRO with the binding affinity 8.4, and ACE2. There are 2 compounds having the same value. There are 8.2. From this result, we know that all the phytochemicals could interact with all target proteins. It means that it shows an anti-coronavirus activity. The phytochemicals which is able to interact with 3-CL-PRO and PL-PRO indicated that they have inhibition activity of coronavirus replication. And the phytochemicals which is able to interact with ACE2 indicated the inhibition activity of the entrance of coronavirus into the host cell. This is the 2-dimension graph of the interaction between the compound and the protein. This is the interaction between myricetin and 3CL4. We can see that there are 7 hydrogen bonds formed. There are glutamine 166A, 7144A, glycine 143A, alanine 145A, threonine 25A, and cysteine 44A. This is ribonectin, showing interaction with PL4 due to formation of 3 hydrogen bonds. There are arginine 66P, threonine 75P, and valine 58P. And this is myricetin and ribonectin showing the same interaction level. Of course, they also form the same number of hydrogen bonds. There are 5 hydrogen bonds here. There are glutamic acid 489B, leucine 475B, leucine 675B, and histidine 493B. We conclude that ISIBIO cultivated in Purwodadi Garden contain phytochemicals with potential of anti-coronavirus activity based on in silico study. Of course, this is only an initial screening. We still need to confirm this effectiveness using further researches both in vitro and in vivo. We also confirm that this potential activity of ISIBIO become the reasons that amplifies the reason for in situ and ex situ conservation of ISIBIO. That's all we have, and we thank you for the attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Mrs. Elo, for a very nice presentation and also very good idea to extract some hardwood bujing. Okay, insiluta bujinga. Okay, so first I invite again number 24, or maybe the committee can apply the video presentation for the presenter number 24.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. First of all, allow me to thank you for the time given to me. And the second time, let me introduce myself. My name is Rizamel from Faculty Agriculture University. On this occasion, I will present a paper entitled Biodiversity Automatic System, Prototype Automatic Fertilization System Integrated Biotech on Internet of Things. Human urine is one of the primary waste in urban areas. Healthy people can produce as much as 1 to 1.3 liter of urine per day. Human urine was in Indonesia has not been maximally processed and utilized. People prefer to dispose of their urine directly into a septic tank or directly into the river waters than having to process it. Human urine itself can be processed into liquid organic fertilizer, LOF, and at a low cost. Processing of human urine into LOF and carried out using special processing procedure to minimize the negative impact of its use as fertilizer. The activity will be held in June to August 2021 at the Faculty of Agriculture, Tanjung University. The method we use is an experiment that starts with designing a prototype technical design. The process in making a prototype and designing software. In the processing of design the prototype, it is done by making an initial design of the prototype design based on the reference that have been obtained. The process of making the tool is carried on the design that has been made, where microcontroller and sensor devices are connected to each other. Then the program is made according to the reference obtained. When the program is made according to the design, then process will be manufactured on process tank stand and solar panel. After all the devices have been put together, then testing the tools. Well, this is from a tool in a 3D image consisting of the microcontroller system, solar panel system, urine processing system, and fertilization. And this blend application which consists of several information, namely fermented urine, pH, depth, soil moistures, cloth height in tank, time, and fertilization. The automatic fertilizer flow in the Biotechnology automation system can be seen if figure. The biotech is connected to a urine reservoir for the incubation process acids by microorganisms from EM4. The incubation phase is connected to a pH sensor to detect the minority level of biofertilizer. The water pump delivers biofertilizers to the biofertilizer reservoir automatically, and the incubation chamber is designed to be closer to prevent ammonia from evaporation and causing unpleasant odors. Incubation is stretched out until the pH is above 9. After the pH sensor shows the urine pH reaches 9, the water pump automatically turns on the distribute the urine from the incubation to the biofertilizer reservoir. And the biofertilizer cycle can also connect by a fertilizer feed to the agricultural plant directly. This tool works automatically using field and clock accuracy detection and sensor with the NAP MCO ESP 2H66 microcontroller to control the time and duration of blood flow amplification to the flow. However, controlling and fertilization time duration of this tool can also be done using Internet of Things technology, which can be controlled via a smartphone application. Testing the automatic dispensing of fermented love to the pinole stone tanks. This test aims to prove the accuracy of the time sensor that has been set with the microcontroller. The microcontroller will automatically turn the water pump when its heart races and predetermined time limit of 6 days. HPA can cause the breaking of hydrogen bond that serves to hold DNA standard together. This disruption of the hydrogen bond destroys activity, causing the naturation and death of bacteria. The test was carried out with a 12 trial. The test result in table 1 show that the tool works well. Common urine that has been fermented in the strong tanks flow automatically into the loft tank (LOF tank), leading to the usage every 6 days according to the setting set on the device. This tool will not work if it doesn't match the time set on the device. Automatic fertilization testing count.mco program ESP 2H66. The purpose of this test is to prove the accuracy of the fertilization time that has been set with the automatic fertilization program carried out by this device. The first solenoid valve will automatically open for 2 minutes to drain the fertilizer into the crop valve on a weak sense. The less fertilization and soil moisture is detected 70%. The test was carried out with 12 trials. The results of the automatic fertilization test in tables 2 show that the tool is running well. The first solenoid automatically opened to flow liquid organic fertilizer to the plants according to the time that has been set on the tool. Liquid organic fertilizer will only flow plant automatically once a week, namely on Monday at 16:00 Eastern Indonesian Time, other than the specific time to 12 doesn't turn on. The Biodiversity auto conclusion, the Biodiversity automation system is equipped with application to maintain temps, humidity, and pH. Communication between the system and the brain application is two-way memory. Communication from the brain application to the system, the networks order, and the communication from the system to the brain application, sensor reading data. The result can process it into the biofertilizer and fertilize automatically 95% based on regulated condition and can be monitored with application via a smartphone. In the future, it is this tool will carry out several improvements such as packaging, according to condition in various agricultural locations, and also adding security system to applications such as username and password, so that control is not quickly taken over. Thank you.
Thank you very much for your nice presentation, Mr. Rizamel, and then for all the speaker and also participant. Now we move into the question and answer session. So please turn on your camera and your microphone for the presenter. So in chat box, we already have 3 questions and all was subject to Mrs. Urip Perwitasari. So the first question was from Firda Dimawarnita from Vivi BBI. The question is, the ethanol has a toxic character and the citric acid is widely used in food industry, and how could this problem be solved? So please, Mrs. Urip, for your question.
Thank you for the time. It is clear, my voice?
Sorry?
It is clear?
Yeah, clear.
Okay. The ethanol in lower concentration in medium can increase the permeability the cell. So the cell can secrete the citrate from the cell into the medium. And for the purification citric acid from media, we can recover citric acid by precipitation or maybe by extraction. I think that's for Ms. Firda.
Okay, and the second question was from Ms. Penny Lestari from BRIN, also to Ms. Urip Perwitasari. So the question is, there are different results of citric acid content on cocoa pod hulls that produced by your present result and the preview result. Can you explain the factor influence in this result?
Okay, thank you, Ms. Penny. In our previous research, we did solid fermentation for the technique fermentation. But the time incubation for the fermentation is quite too long, which is 10 days incubation. And many studies have report that submerged fermentation can short the fermentation time. So we tried submerged fermentation to make a short time incubation.
Okay. Are you clear, Bu Penny, with the answer?
Yeah. Thank you, Ms. Orip. But I see that in previous result, it have higher 7.5, if I'm not wrong, than the present result, only 1.5. So I just think that your previous method is better to extract the citric acid than this present result. How do you what? Apa pendapatmu lah?
Yes. It is it is because the cacao potash is the significant factor for the citric acid production. It is shown in the ANOVA that show cacao potash is the significant factor because that is substrate in submerged fermentation only 1% than solid fermentation. That's why the citric acid yield is lower than solid fermentation.
Oh, okay. Yeah, thank you. And thank you for the moderator for the time.
Okay, if you have any question, maybe you can ask via email or something, Bu Penny. And the third question, Mrs. Urip from Mr. ISIBIO. From your result, it can be seen that only cocoa potash has significant effect in the citric acid yield. So which other variable were not significant? So it suspect that the concentration ring of the two letter variable is far away from the optimum value of citric acid. So did you do the preliminary experiment by using the two-level factorial with the central point to observe the significance of the selected concentration? So I think this question is more to the research design concerning the concentration used in the methodology. So
Okay. Thank you. It is related with Miss Penny. Actually, this is our preliminary study for citric acid production from cacao potash by submerged fermentation. The factors and the range we use in this research is based on the previous literature. It is said that nitrogen and time incubation is a factor that affecting of citric acid production. That's why we use the factor and the range. However, further research are needed to find another factor that affecting the production of citric acid by cacao potash as a substrate in fermentation. I think that's all. Thank you.
okay, Dr. Bayu, are you in Rome? Is it clear? You have
Yeah.
comments?
Yeah, maybe just suggestion, because usually for us, when we do the response surface method, generally we in the preliminary study we have to screen first whether that variables are significant or not. So usually people conducting the two-level factorial design first in order to select which one, which variable, and then which range that we want to move to the response surface method. Want to know that the optimum value of the variable. So I think maybe I just give suggestion that before you conduct the RSM, if you do the preliminary screening first by using two-level factorial design with center point, maybe you will get better results, in which you will get the optimum value of the variable that you screen by the RSM. Because from here we just see that only one variable which had a significant effect into the citric acid yield, and we cannot say that is the optimum value because you just get one variable. Usually when we do the response surface method, we will have more than one, usually 2 or 3 variable that we have for the optimum experiment. Maybe only that from me, Pak Fahru Rozi.
Okay, thank you, Dr. Bayu, for your comment and your suggestion. Maybe you later we can talk again with Mrs. Urip. So we still have about 3 minutes for the question. So please, to the participant, if you have one question, 1 or 2 questions is still available. So if there is no question, so I will ask to Mrs. Martasari. So in your presentation, you mentioned that from the melinjo, you have a lot of substance, for example, like polyphenol, pectin, and also the other content. So which substance you suggest as antioxidant, have antioxidant activity? And then the second question, in your slide, you show the HPLC result, and then there is some different the peak in your result, but you don't mention what kind of peak in your result. Bu Marta, are you?
Hello. Hello.
Hello.
Yes. Okay. Thank you for your question. I will try to your question. The first question is in the bioactive compound from the seed melinjo contain the some of the stilbenet compound consisting and the gnathin, sperator, and so on, and in this study, it's preliminary study. I just focus this research to investigate the antioxidant and the profile and the interpretation, the data from the sample without having the standard. Okay. And in this, we want to view the profile HPLC profile chemical compound by HPLC, and to interpret the data, interpretation the compound from the GCMS. Maybe its studies can be future developed. Need to some
purification to know that who is the responsible as the bioactive compound to give the good activity and the perform the bioactive compound.
Okay, thank you, Mrs. Marta, for your answer. I think it's clear, but I think Dr. Bayu should suggest to Bu Marta maybe should use internal standard, because we have also internal standard. I think maybe it's some polyphenol, I think, but I don't know exactly. Okay, thank you, Bu Marta, for your answer. And then if there is no question, I will ask one more question to Mrs. Elo. Do you, I don't know exactly, maybe I'm not mistaken. Do you extract the protein from Insia bijunga, or you just literature study the protein was EBC or something like this, or you extract by your own?
Thank you for the question, Mr. We do not extract the compounds. We only check the compounds through heteroscopy. We found 6 compounds there. And by using the 6 compounds, we integrate those compounds with the proteins involved in coronavirus infection. That's all. It's all we called it a dry lab with no extraction process at all.
With lab, yeah, okay. So, but for example, if you have active compound from this wood, I think I said this is in Indonesia we called marbau wood.
Merbau, yes, merbau, sir.
Hardwood. So, if you find something, so how we can extract from? Do you have idea how we can extract from this wood, because it's a hardwood?
Yes, but we found out that the compound is contained not in the hardwood but in the bark, sir.
Sorry, in what?
Bark, kulit batang.
Oh, batang.
Kulitnya, bark, kulit batang.
Oh, kulit batang. Okay.
Yes.
Okay.
In previous to continue the study, actually we have the idea to extract the bark, but this is collection. It's hard. Karena ini koleksi jadinya susah gitu.
Oke. Ya, terima kasih ya Bu Elo atas jawabannya.
Thank you.
Okay, I think we passed the time for the organizing committee, and then I don't need to summarize all of this presenter. And then thank you for your attention, for all of your participants, and also for presenter. See you next year, and I will get back to organizing committee. Thank you.
Thank you very much to Dr. Fahru Rozi and to all presenters for participating in this session. We expect you to join us at the main room because we have some announcements and for a closing session. Thank you.
Recording in progress.
Ladies and gentlemen, welcome back to the main room of ISIBIO 2021. Before we waiting for others to join us from other breaking up room, we would like to remind you that for the second day of ISIBIO tomorrow, we will open the registration at 7:30 and start at 8 a.m. Jakarta time. And for tomorrow, we have keynote speaker Professor Jugdeo from Asian Federation on Biotechnology, and plenary session we will have speakers Professor Zihua Jiang from Department of Animal Sciences, Washington State University, USA, and Mr. Zahir Ali, PhD, from Center for Desert Agriculture, King Abdullah University of Science and Technology, Saudi Arabia. And at last, there will be announcement of 6 best presenter in ISIBIO 2021. So stay tuned and we hope to see you tomorrow. Thank you. Ladies and gentlemen, welcome back to the main room of ISIBIO 2021. We would like to thank all the speakers and moderator for the insightful session. Now we have come to the end of the first day of the 8th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering, ISIBIO 2021. We would like to remind you for the second day of ISIBIO tomorrow, we will open the registration at 7:30 and start at 8 a.m. Jakarta time. And for tomorrow, we will have keynote speaker, Professor Duk Jo from Asian Federation on Biotechnology. And plenary session, we have speakers, Professor Zihua Jiang from Department of Animal Sciences, Washington State of University, USA, and Mr. Zahir Ahli, PhD, from Center for Desert Agriculture, King Abdullah University of Science and Technology, Saudi Arabia. And at the end of the symposium tomorrow, there will be announcement of 6 best presenter in ISIBIO 2021. So stay tuned, and we hope to see you too tomorrow. We would like to thank all of you for your participation. And on behalf of the official committee, we would like to apologize for any inconvenient matters during this today event. So see you tomorrow and goodbye.